The ocean is quiet in the way only open water can be quiet. A vast, weightless stillness where every drifting organism, every faint current, every suspended flake of life becomes part of an endless blue tapestry. Into this calm drifts a body. It is a sea turtle, or what remains of one, suspended just below the light-drenched surface like a forgotten relic. The shell is cracked, the edges softened by the beginning stages of decay. Thin strands of skin sway like loose ribbons in the passing water, and a faint cloud of oils and microscopic tissue spreads outward in a barely visible haze. It is the kind of sight that marks the closing chapter of one life, but, in the strange arithmetic of the ocean, also signals the beginning of a cascade of biological activity that will ripple through the ecosystem for days. The camera holds steady on the carcass, and the blue behind it deepens into a darker gradient. Something stirs in the shadows, far beyond the edge of clear visibility. At first, it is only a shift in background texture, an almost imperceptible disturbance of the water column. But as it moves closer, the shape hardens into the wide, unmistakable silhouette of a tiger shark. The animal does not rush. It glides with the unhurried confidence of a predator that has perfected the art of energy conservation over millions of years. Each movement is deliberate. The tail sweeps once, twice, propelling the massive body forward with astonishing efficiency. The shark approaches the carcass like a patrol vehicle rolling up to a stalled machine on a deserted highway, not as a hunter chasing prey, but as a specialist responding to an inevitable part of the ocean's cycle. A dead animal in the water broadcasts a chemical signal that spreads in all directions. Long before the tiger shark sees anything, it smells it, thanks to sensory adaptations that allow the species to detect minute concentrations of amino acids, fats, and decomposition byproducts from many kilometers away. These signature compounds ride on currents like invisible smoke trails. Where most fish would detect only vague shifts in background chemistry, the tiger shark reads the water like a map. The nose interprets concentration gradients, stronger on the left, weaker on the right, stronger again after a change in current. Each variation narrows the path, guiding the shark into what functions as a natural search track. This is not wandering. It is methodical navigation, a chemical pursuit perfected through evolutionary pressure. As the shark draws closer, small fish scatter from beneath the drifting turtle. Some are juvenile jacks and wrasses, opportunistically picking at softening tissue. Others are cleaners that inspect anything that appears still enough to host parasites or bits of flesh. They all know when to leave. The tiger shark enters the clearing around the carcass like an aircraft entering restricted airspace. The smaller fish peel away instantly, responding to the shift in behavior from all life around them. The shark circles once, passing the carcass with slow, cautious precision. Tiger sharks are often portrayed as reckless feeders, but their approach to matter that is already dead is almost analytical. They test the water with taste receptors embedded in the lining of the mouth, gauging the age of the carcass, the state of decay, even whether another scavenger has already fed here. The shark tilts slightly, lowers its head, and finally makes its first contact. The jaws open not in a dramatic strike, but in a steady, deliberate bite that cracks through softened shell and sinew. When the shark pulls back, a plume of fragments scatters in the water. Tissue, bone dust, oils, and microscopic debris that form the first release in a chain of nutrient redistribution. It is here that the role of the tiger shark becomes clear. This is not just a predator. It is a sanitation force, a mobile decomposition system built into the ecology of coastlines and open water. The shark tears another piece from the turtle, and with that movement, accelerates the breakdown process far beyond what currents and bacteria could accomplish alone. Large predators fragment carcasses into sizes that smaller scavengers can handle. Loose chunks drift downward, settling on reef outcrops or sinking to sandy bottoms, where crabs, brittle stars, and worms wait for whatever fortune delivers. Microscopic organisms cling to suspended particles and begin feeding immediately. In this way, one dead turtle becomes a decentralized feast, feeding every trophic tier from sharks to bacteria. If this shark had not arrived, if scavengers like it did not exist, coastlines would be graveyards. In regions where large animals migrate seasonally, such as turtles moving toward nesting beaches or dolphins traveling across shelves and lagoons, natural mortality produces a steady flow of bodies. Without the intervention of tiger sharks and other scavengers, these carcasses would accumulate in bays, estuaries, and shallow reef systems. Rotting flesh consumes oxygen as it decomposes, and microbes responsible for the breakdown proliferate rapidly, creating zones of depleted oxygen that suffocate fish, invertebrates, and even corals. The ocean has a self-cleaning mechanism because it must. A system built on constant cycling cannot afford stagnant pools of decay. Tiger sharks, along with other scavengers, are the frontline workers preventing such ecological collapse. The shark takes a deeper bite now, wrenching part of the body free with a twist of its massive torso. The movement sends a shockwave through the water, and the freed tissue drifts downward in slow, spiraling motion. The camera follows it toward the seafloor, showing how each fragment begins a secondary phase of decomposition ecology. Crabs emerge from crevices, small fish dart in, taking nervous nips. Microbial mats begin to bloom on surfaces rich in lipids, forming tiny biochemical hotspots. These sites, though temporary, fuel population pulses in small scavengers and filter feeders. In regions where carcasses are frequent, the presence of tiger sharks directly shapes community structure by accelerating the rate at which nutrients are released and redistributed. Up above, the shark continues feeding with the calm persistence of an animal built for endurance, not frenzy. Tiger sharks have exceptionally broad diets. Their teeth, serrated, recurved, and triangular, are engineered to slice through almost any marine tissue. They can shear through turtle shells, break into dolphin rib cages, and disassemble decaying whales that drift into coastal waters. Their digestive systems tolerate a wide variety of organic material, even bone fragments and cartilage. This tolerance allows them to serve as apex-level scavengers, capable of processing the toughest remains in the ocean. It is not just physical adaptation that makes tiger sharks ideal carcass specialists. They occupy a unique spatial niche. These sharks regularly patrol coastlines, longshore currents, and migration corridors, where drifting bodies are most likely to accumulate. Their wide-ranging behavior, covering many tens of kilometers in a day, increases the probability that they encounter carcasses soon after death, before decomposition reaches stages that become harmful to surrounding ecosystems. In this way, tiger sharks function as highly efficient search and response units that activate whenever the ocean signals that something has died. The camera shifts perspective, pulling back to reveal the surrounding environment. To the untrained eye, it looks empty. But the water is full of chemical trails, dissolved nutrients, drifting fragments, and signals that carry meaning for every species tuned to read them. The presence of a carcass does not simply attract scavengers. It reorganizes the entire micro-ecosystem around it. For hours after the tiger shark finishes feeding, smaller predators and invertebrates will arrive. The site will become a small but intense hub of activity, a brief eruption of life fueled by one death. And at the center of this process is the shark, the first responder, the animal that transforms a single body into an ecological event. The importance of this role becomes especially obvious along coasts with large populations of marine mammals or turtles. Every year, natural mortality creates significant biomass that would otherwise sink into deep channels or drift into fragile habitats. By consuming carcasses early, tiger sharks regulate not just the physical cleanliness of the sea, but also the microbial dynamics of coastal waters. They reduce the spread of bacteria that flourish in decomposing flesh and help prevent imbalances that could trigger disease outbreaks among fish and invertebrate populations. Their presence helps maintain stable oxygen levels in shallow lagoons where decomposition can quickly create dead zones. In many ways, they operate as biological regulators, ensuring that the ocean's recycling system stays efficient. The shark now tears the carcass almost in half with a final decisive bite. The camera closes in on the jaws, revealing details that often go unnoticed. The rows of replacement teeth forming behind the primary line, ready to rotate forward when the front teeth wear down. The thick pads inside the mouth that protect the shark from sharp bone edges. The powerful musculature that locks the jaws into position. When the shark pulls away, the body finally falls apart completely, sending the last of the solid pieces drifting downward. The tiger shark pauses, then turns its head slightly as if testing the water once more. It might detect distant signals, another carcass, a wounded fish, or simply a shift in current that suggests a new direction to patrol. With a slow, controlled motion, the shark glides away from the scene. The water behind it fills with the first returning fish that dare approach the remains. Within minutes, the larger fragments are claimed by smaller predators, and within hours only trace elements will remain, absorbed into the sediment, the plankton, or the bodies of countless small organisms. This is the quiet, unseen service tiger sharks perform. They do not hunt for glory or spectacle. They work in the shadows of the ocean's operating system, ensuring that nothing is wasted, and that the sea does not become overwhelmed by the byproducts of its own vitality. They are not just hunters. They are custodians of the coastline, enforcers of ecological hygiene, breakers of decay, and guardians of water quality. Their movement through the ocean is a constant reminder that life and death are not separate states here. They are phases in a continuous cycle that depends on predators capable of managing both. When the camera pans back to the open blue, the turtle's remains are already fading into the distance. The water looks unchanged, as if nothing significant occurred. But beneath that illusion is an entire cascade of activity set in motion by a single scavenger. In the days that follow, nutrients from this one carcass will pass through dozens of species, fueling growth, reproduction, and the microscopic engines that keep the ocean breathing. All because a tiger shark arrived on time, as it has for millions of years, to do the work that holds marine ecosystems together. Along the rugged, temperate coasts of the world, especially where cold currents surge upward from the deep, the story of life unfolds with a harsher rhythm than in the tropics. Here, waves crash against basalt cliffs, kelp forests sway like slow-motion cathedrals, and the wind carries a constant smell of salt, algae, and distant decay. The ocean feels heavier, richer in nutrients, and far less forgiving. In these environments, massive colonies of seals gather every breeding season, turning entire stretches of shoreline into teeming, chaotic cities of barking adults, squirming pups, and relentless competition for space. Life here is loud, crowded, and physically brutal, and it naturally produces another constant: death. The camera opens on this shoreline. Young seals jostle between aggressive males, some drowning in frantic pile-ups at the surf line, others weakened by malnutrition or injured during territorial clashes. Predators, storms, and disease take a steady toll. During peak breeding months, the colony churns out carcasses at a pace that would overwhelm any ecosystem lacking a powerful cleanup force. The beach becomes a conveyor belt of mortality. Dead pups are tossed back into the waves by incoming tides. Weak individuals collapse at the water's edge, drifting into the surf as soon as they lose their grip on the rocks. Behind the noisy theatrics of the colony lies a darker, quieter truth. Seal nurseries create graveyards as much as they create life. The footage follows a single carcass now, a pup, perhaps a few months old, its body limp as the surf lifts and drops it repeatedly. A swell finally pulls it past the breaking waves into a calmer stretch of green-blue water. It tumbles in lazy rotations, limbs extended, patches of fur peeling away. The body is no longer just a victim of the colony's harshness. It has become a signal that radiates outward through the water column. The decomposition process begins immediately, releasing amino acids, fats, blood, and gases. These chemicals disperse through the current like a complex, multi-dimensional scent trail, one that only certain predators can interpret with accuracy. Far offshore, beyond the visible reach of even clear coastal water, the outline of a great white shark comes into view. It is cruising along a predictable path parallel to the coastline, moving with slow, economical strokes of its tail. Great whites do not wander aimlessly. Their movements are guided by experience, instinct, and a set of sensory adaptations unmatched in the marine world. The shark's nostrils detect the faint chemical stream from the seal carcass long before its eyes register anything. Slowly, the shark adjusts its trajectory, turning toward the coastline with the confidence of an animal that has made this journey many hundreds of times. As the shark moves closer, the camera reveals subtle details of its anatomy that explain why this species is not merely a hunter of healthy prey, but also one of the ocean's most efficient scavengers. The great white's olfactory system contains specialized folds that dramatically increase surface area, allowing it to detect incredibly low concentrations of organic compounds. Its lateral line senses minute vibrations, picking up the tumbling rhythm of the drifting carcass long before it enters visual range. And its electroreceptive organs, the ampullae of Lorenzini, can detect the faintest electrical gradients. Although a dead seal emits no heartbeat, the shark still interprets anomalies in temperature and density around the body. Many people imagine great whites solely as high-speed predators chasing seals in explosive bursts of power, but the reality is more nuanced. These sharks routinely patrol areas around seal colonies, not just for hunting opportunities, but for scavenging. The constant availability of dead or dying seals provides a dependable energy source, far easier to access than sprinting after a perfectly healthy adult. Evolution rewards efficiency, and a shark that conserves energy while consuming high-quality protein is one that thrives. This behavior is not a fallback strategy. It is a central part of the species' ecological role. The drifting seal finally enters the shark's field of vision. The great white slows, its massive form stabilizing in the water with minimal fin movement. Scavenging requires careful inspection. A dead or dying seal can carry disease, but to the shark that does not matter. Pathogens rarely cross the evolutionary gap between mammals and fish. What the shark is actually analyzing is how recently the animal died, how much energy remains in its tissues, and whether other scavengers have already begun feeding. The shark passes the carcass once, slow and deliberate, the way a vulture might circle above a fallen animal on land. Then the shark turns slightly and takes its first bite. The jaws of a great white are engineered for power rather than finesse. Cartilaginous supports, massive muscle anchors, and serrated teeth combine forces to carve through thick blubber and underlying muscle. But even during scavenging, the shark does not frenzy. Instead, it takes a controlled, calculated bite that slices a substantial portion from the carcass. Great whites often feed this way on floating remains. One heavy bite, then a moment of stillness as the animal considers its next move. This method prevents wasted energy and reduces the risk of losing caloric gain through unnecessary struggle. Fragments of seal tissue drift downward, forming a thin snowfall of organic material that instantly attracts smaller scavengers. Kelp, bass, rockfish, and even amphipods rise from the forest floor below, taking advantage of this brief nutrient windfall. This process repeats around every carcass consumed by great whites along these coasts. The sharks initiate the breakdown, but countless other organisms complete it. Without this top-level fragmentation, many carcasses would rot intact for days, releasing large localized bursts of decay that stress the surrounding environment. The narrative broadens now to show the colony from a wider angle. The number of sick or injured animals produced daily varies depending on season, weather, and the density of pups, but across major seal rookeries worldwide, the mortality rate is high enough to create a constant stream of bodies. Great whites capitalize on this supply, returning year after year to the same beaches, bays, and offshore channels. The sharks act as mobile filters that remove weak genetics, diseased animals, and the natural casualties produced by overcrowding. In doing so, they reduce viral and bacterial load within the colony. A population without scavengers would suffer from a buildup of rotting matter that could contaminate nurseries and increase disease transmission. This dynamic creates a surprising relationship between predator and prey. Although great whites hunt seals, they also serve the colony by removing individuals that would otherwise become sources of infection. This indirect benefit stabilizes the population over time. In ecosystems where top predators decline, due to fishing, habitat degradation, or intentional culling, the absence of this natural pressure leads to imbalances. Seal colonies expand beyond the carrying capacity of their coastline, competition intensifies, waste accumulates, and disease outbreaks become more common. The great white's presence, though feared, is a regulatory mechanism rooted in evolutionary logic. The story shifts deeper now, away from the surf zone and into open water. Here, the camera reveals a colossal form drifting like a sunken continent. The carcass of a whale. Even far offshore, dead whales create immense ecological events known as whale falls, providing habitats that can last decades in the deep sea. But along temperate coasts, when such carcasses originate in shallower waters, great whites often arrive early in the decomposition process. A whale's blubber is one of the richest energy sources available in the marine world. A single scavenging event can sustain an adult great white for weeks. Sharks approach whale carcasses cautiously. The first bites open the thick skin, releasing a flood of oils into the water. The surface becomes streaked with shimmering patches that glisten in the sunlight, drawing in other scavengers, both sharks and fish, from many kilometers away. Great whites position themselves strategically, often along the flanks or near the tail where bone structure is easier to access. These feeding events serve as massive nutrient redistribution episodes, accelerating the dispersal of energy from the whale body into the broader food web. Returning now to the coast, the camera follows another seal drifting into deeper water. The seal's movements indicate severe sickness, jerky, uncoordinated motions that betray neurological dysfunction, possibly from parasitic infection or toxin exposure. Many seals die this way, succumbing to conditions that spread rapidly through dense populations. Great whites are finely tuned to detect these moribund individuals. Their sensory systems interpret erratic swimming behavior, and their electroreception picks up weak, inconsistent muscle signals. Removing these animals before they die naturally prevents their bodies from decomposing within the colony, where bacterial loads could spike quickly. The shark strikes decisively, ending the animal's suffering instantly. This behavior is often misinterpreted as predation alone, but ecologically, it is closer to targeted removal of sick individuals. Over long periods, this selective pressure favors healthier genomes within the colony. Evolution rewards seals that can grow faster, avoid injury, and resist disease. Traits that indirectly benefit the entire ecosystem. The camera widens again to reveal the coastline in full. Waves carry bodies out, sharks move along predictable patrol routes, and smaller scavengers feed on the remains unearthed by these apex predators. It is a system built on constant turnover, where life and death are tightly interwoven. The great white shark is not a roaming menace, but a necessary component of this cycle, preventing the seal graveyard from overwhelming its own waters. Without these sharks, carcasses would accumulate in the surf until storms or currents dispersed them. Bacterial blooms would erupt in stagnant areas, decreasing oxygen levels and killing fish that rely on well-oxygenated water. Entire coves would periodically become anoxic, creating dead zones that ripple through surrounding ecosystems. The perception of great whites as simple hunters obscures their true ecological role. They are regulators of population health, stabilizers of coastal environments, and the primary agents preventing the buildup of decay in some of the most productive waters on Earth. The chapter closes on a final image. A great white cruising away from the colony, its body moving with slow, confident precision. Behind it, the coastline continues its noisy chaos. Newborn life, inevitable death, and the steady rhythm that ties the two together. The shark fades into the green, returning to its patrol, ready to respond to the next signal drifting down the current. It is the unseen custodian of the seal graveyard, maintaining an ecological balance that would collapse without its presence. The open ocean at first glance appears deceptively simple. Its surface is a shimmering plain, interrupted only by the rise and fall of long swells and the intermittent breath of distant animals surfacing for air. But beneath this shifting skin lies a vertical world so vast and layered that even the largest creatures on Earth vanish into its immensity. In this expanse, life and death unfold on a colossal scale. And nowhere is that magnitude more obvious than when the camera descends upon a pod of orcas moving deliberately across the blue horizon. Their course is purposeful. Their formation is tight and coherent. They are not wandering hunters. They are specialists, with a target so immense, it reshapes the very chemistry of the ocean when it dies. The opening scene centers on a mature baleen whale surfacing for air. It exhales with a thunderous plume of mist, the sound rolling over the water like a drumbeat. This individual may have traveled thousands of kilometers during migration, surviving storms, parasites, and the strain of reproduction. But today, the whale is not alone. Several black and white forms arc just beneath the waves, their dorsal fins slicing the surface like the keels of fast-moving boats. The orcas are silent at first, assessing the whale's pace, its breathing rhythm, and any signs of weakness. They attack only rarely, and only with a strategic plan honed by countless generations of learning. The encounter begins with a slow tightening of the circle. Orcas are members of the dolphin family, and their intelligence is expressed not only in problem-solving, but in communication so complex it blurs the line between instinct and culture. Different pods use different hunting strategies passed down through mothers and grandmothers, with some specializing in fish, others in seals, and a select few developing the astonishing ability to bring down whales many times their size. The pod here belongs to the latter. They approach not as reckless predators, but as coordinated tacticians. The first strike is not a dramatic bite, but a disruptive maneuver. One orca rushes at the whale's flank, causing it to lurch sideways and expend precious energy. Another swims ahead, positioning itself near the whale's rostrum, forcing it to alter course. The calf-sized individuals stay back, observing every motion as if absorbing a lesson. The adults work in shifting roles, creating confusion, inducing panic, and wearing down the whale's stamina. Baleen whales are powerful but not agile. In these tight spirals, endurance becomes their only defense, and endurance drains quickly. The fight intensifies when the orcas begin targeting the whale's blowhole. Each time it surfaces to breathe, they lunge forward, not to kill instantly but to obstruct its inhalation. This forces the whale back under sooner than it intends, extending dives beyond comfortable limits. A whale deprived of oxygen becomes clumsy, and clumsiness in the open ocean is fatal. Eventually, the animal slows, its tail beat weakening. The orcas seize the opportunity. One breaks formation, accelerating beneath the whale's underside and ramming hard into its jawline. Another clamps onto the soft tissue near the pectoral fin. Together, the pod overwhelms the giant, not with brute strength alone, but with relentless teamwork. The whale succumbs. Its final exhalation escapes in a burst of bubbles as its immense frame slackens. For a moment, the scene is silent. Even the orcas pause, as if acknowledging the magnitude of the life they have just ended. The adult females swim close, guiding younger individuals toward the carcass to teach them where to feed. The best cuts, the tongue, the lower jaw fat, the internal organs, are consumed first. These parts offer unparalleled caloric density, rich in lipids and essential nutrients that sustain the pod through extensive travel. The orcas feed selectively, not devouring the entire whale, but removing strategic portions that maximize nourishment for the least energy expense. As they tear into the flesh, large chunks drift away. Blood mixes with seawater, turning the surface into a mottled red field that quickly attracts other predators. Within minutes, the first scavenging sharks arrive. Blue sharks, makos, and even opportunistic great whites. They dart in cautiously, snatching loose pieces while staying clear of the orca's powerful jaws. Although these sharks could never kill a whale alone, they benefit enormously from the orca's work. The initial feast is a chaotic process, filled with swirling bodies, suspended tissue, and a flurry of movements that echoes the energy of a savanna carcass mobbed by hyenas and vultures. But this spectacle at the surface is only the beginning. After the orcas take their preferred portions, they eventually move on, leaving behind a whale body that is now buoyant with decomposition gases. The carcass drifts, carried by current and wind. For hours or even days, it may remain near the surface, serving as a buffet for seabirds, sharks, and fish. Gradually, the body becomes waterlogged, its density shifts, the gases disperse, and the carcass begins its descent into the abyss. This slow sinking is one of the most dramatic transitions in marine ecology. A massive transfer of nutrients from surface ecosystems to a realm where food is scarce and life must take what it can get. As the whale sinks deeper, pressure increases and light disappears. The body becomes a silhouette, then a shadow. then nothing more than a faint suggestion of shape against total darkness. When it finally touches the seafloor, often at depths measured in thousands of meters, a transformation begins that is unmatched in scale by almost any other natural event. Orcas, by taking down large whales, create these whale falls, ecological islands that fertilize dead zones of the deep for decades. The first organisms to arrive are scavenging fish and deep-sea sharks. Hagfish and sleeper sharks slither and glide in from the darkness, drawn by chemical cues. They feed aggressively, burrowing into the carcass and stripping away remaining flesh. Crabs swarm in next, cracking through cartilage and picking at connective tissue. This initial scavenger stage can last weeks, reducing hundreds of kilograms of whale tissue into loose fragments that disperse over a wide radius. Following this phase, bone-crushing specialists take over. Amphipods, tiny shrimp-like scavengers, consume softer tissues lodged between bone surfaces. They reproduce in massive numbers around whale falls, doubling or tripling in local density due to the sudden abundance of food. Entire generations may live and die on the whale's remains. Then begins the enrichment opportunist stage, where organisms that are not specialized scavengers but opportunistic feeders colonize the site. Worms, snails, and small crustaceans burrow into nutrient-rich sediments beneath the bones. Some species appear only when whale falls occur, disappearing afterward until the next arrival. Their presence reveals how many deep-sea organisms rely on rare pulses of food descending from the surface. But the most unusual stage is the final one, the sulfophilic stage, which can last for many decades. Whale bones rich in lipids slowly decompose anaerobically, releasing hydrogen sulfide. This chemical, toxic to most animals, becomes the energy source for chemosynthetic bacteria. These microbes metabolize sulfur compounds the way plants metabolize sunlight, creating a foundation for an entire miniature ecosystem. Tube worms, clams, and other organisms thrive on the microbial mats. The whale fall becomes a long-term habitat, a self-sustaining island of life in an otherwise barren desert. This entire cascade, from orca kill to deep-sea microbe colony, begins with the dramatic event at the surface. Orcas are not simply hunters of giant prey, they are catalysts of ecological redistribution on a planetary scale. Every whale fall begins with predation or natural death, but predation by orcas increases the frequency and geographic spread of these events, especially in regions where whale populations migrate seasonally. Their role ensures that the deep sea, which receives remarkably little organic material, periodically receives nutrient jackpots capable of supporting life far beyond what normal sinking particles could provide. The importance of this process is difficult to overstate. The deep ocean is Earth's largest ecosystem, but it is also one of the most nutrient-poor. Without whale falls, many species of deep-sea invertebrates would struggle to persist. Genetic studies reveal organisms so tightly linked to whale falls that their life cycles synchronize with the presence of these enormous carcasses. When orcas kill whales, they are not merely consuming them. They are setting in motion ecological events that resonate through centuries. The chapter closes by returning to the pod of orcas now traveling steadily across open water. They leave no evidence of the colossal transformation they have initiated. The sea around them looks unchanged, yet far below, in darkness beyond reach of sunlight, a whale lies on the seafloor feeding an entire hidden world. In this way, orcas sculpt not only the living landscape, but the dead one. They forge islands of decay that blossom into islands of life. They are not just predators, but engineers of the deep, architects of rotting giants that nourish the abyss for generations. The deep ocean begins long before the light fades. At the surface, the world feels deceptively alive and familiar. Sunlit water flecked with spray, seabirds patrolling the air, and small fish scattering beneath shifting patches of foam. But just a few tens of meters down, brightness dissolves into muted green, and then, within a few hundred meters more, into total darkness. Below this boundary lies a realm shaped by pressure, cold, and scarcity, a vertical wilderness where almost nothing moves without purpose. Into this dark column descends a creature built to navigate its depths with confidence, the sperm whale. The descent begins with a single inhale. A sperm whale draws in air with a force that expands its immense ribcage, folding the lungs into a compact shape suited for extreme pressure. Its blood becomes a reservoir of oxygen, stored in specialized proteins that bind and release gas slowly. With one powerful kick of the flukes, the whale sinks, leaving the glittering surface behind. Sunlight narrows into a white coin overhead, then disappears altogether. The whale's body becomes a moving mass of pale grey against a backdrop of nothingness. This species is the largest toothed predator on Earth, and every dive takes it into a hunting zone where the rules of life are inverted. Eyesight is almost useless here. Instead, the whale relies on the ocean's most sophisticated biological sonar. Its massive forehead houses an intricate acoustic system capable of producing high-intensity clicks. These sounds travel through water, bouncing off squid, fish, and drifting particles. The returning echoes paint a three-dimensional map of the deep. The whale interprets each reflection with extraordinary precision, detecting the flutter of squid fins or the tumbling descent of gelatinous creatures. The prey it seeks is formidable in its own right, large squid species that thrive in the blackness. These animals possess sharp hooks, powerful tentacles, and bodies adapted to near-freezing temperatures. Their movements are jerky and unpredictable, propelled by bursts of jetting inertia, but the sperm whale's approach is methodical. It moves through the water column like a deep-sea survey vessel, scanning every meter for signs of motion. When it detects a squid, the whale accelerates abruptly, using its long hydrodynamic body to lunge forward. The mouth opens, revealing conical teeth designed not to chew but to grasp. The squid thrashes, releasing clouds of ink that dissolve instantly in the pressured environment. Within seconds, the animal vanishes into the whale's throat. This scene repeats dozens of times during a single dive. Every capture sends a pulse of nutrients into the whale's body, fueling its next descent. Over a lifetime, a sperm whale consumes an enormous volume of deep-sea prey, shaping populations of squid across vast regions of the ocean. But its ecological influence extends far beyond what it eats. What it returns to the surface becomes equally significant. When the whale rises from its long dive, its ascent transports nutrients from the abyss back to sunlit layers. The whale expels a plume of fecal matter, a reddish-brown cloud rich in iron and nitrogen. These nutrients are scarce near the surface, yet they are essential for phytoplankton growth. In many regions, iron limits productivity more than light does. By releasing deep-sourced nutrients upward, sperm whales act as biological pumps that fertilize the ocean's upper layers. Their presence supports the very base of the marine food web, influencing everything from zooplankton to fish to the large predators that patrol surface waters. In this sense, each dive becomes a vertical exchange between two worlds. The whale carries energy downward in its body, consumes deep-dwelling prey, and then returns with nutrients that stimulate growth in sunlit zones. Few animals on Earth create such a precise loop between ecosystems separated by more than 1,000 meters of water, but the sperm whale's impact does not end with its life. The camera now follows a different whale, one nearing the end of its decades-long existence. As it weakens, its dives become shorter, its movements lose fluidity. Eventually, the whale takes its final breath at the surface and does not descend again under its own power. Instead, gases trapped within the body keep it afloat, turning the carcass into a drifting island. Seabirds land on its back, picking at the softening flesh. Sharks gather, tearing thick strips of blubber. Ocean currents carry the body away from the zone where it died, pulling it toward deeper water. Slowly, the gases dissipate. The body begins to sink. This transition, though seemingly simple, triggers one of the most remarkable ecological events on the planet: the creation of a whale fall. As the carcass descends, pressure compresses flesh, bones, and fluids. The water grows colder. The last traces of light vanish. When the whale finally touches the seafloor, it comes to rest in a world dominated by stillness. Here, food scarcity shapes every organism, and the sudden arrival of a whale, weighing many tens of thousands of kilograms, is catastrophic in the best possible way. The first responders are mobile scavengers. Deep-sea sharks, hagfish, and grenadiers converge from the surrounding darkness, their senses honed to detect even faint traces of organic matter. Hagfish burrow into the carcass, feeding by absorbing nutrients through their skin and consuming flesh with keratinized plates inside their mouths. Sharks shear off pieces of tissue, leaving long, jagged furrows. These predators strip much of the whale's soft tissue within weeks, reducing it to a skeleton draped in remnants of decaying fat. Once the initial feasting subsides, a new community arrives. This marks the enrichment opportunist stage, during which small organisms colonize the sediments around the carcass. Polychaete worms burrow into the nutrient-rich seafloor. Crabs and shrimp pick through remaining fragments. Snails and amphipods bloom in staggering numbers, drawn by the abundance of decomposing material. These opportunists reproduce rapidly, completing several generations before the site's nutrient levels begin to decline, yet the most extraordinary stage is still to come. As oxygen becomes depleted within the remaining fatty tissues, bacteria shift into anaerobic metabolism. They break down lipids trapped inside the whale's thick bones, releasing hydrogen sulfide, a gas toxic to most marine life. But in the deep sea, where sunlight never reaches, organisms have evolved to use chemical energy in place of light. Sulfur-oxidizing bacteria grow on bone surfaces, forming white, fuzzy mats. These microbes become a primary food source for specialized organisms that cannot live anywhere else. Among them are the bone-eating worms known as osedacs. These bizarre animals lack mouths and digestive tracts. Instead, they anchor themselves to whale bones with root-like structures that penetrate deep into the marrow. Inside these roots live symbiotic bacteria that break down fats and convert them into nutrients the worms can use. Each osedac's colony grows into a miniature forest of gelatinous stalks, waving with currents that barely register at the seafloor. The appearance is eerie, an otherworldly garden blooming from the skeleton of a fallen giant. This sulfur-philic stage can last for decades. Over time, the whale skeleton becomes a hub for an entire community of organisms adapted to chemical energy rather than sunlight. When the bones are finally depleted, the site slowly disperses. The organisms migrate onto the broader seafloor or die off, leaving behind only traces of their presence. But the genetic lineages they bolster will persist until the next whale fall arrives. Perhaps many years later, delivered once again by the death of a deep-diving giant. This recursive relationship between life and death defines the sperm whale's place in the ocean. Every dive it completes fertilizes the upper layers through nutrient plumes. Every hunt shapes populations of squid in the depths. Every carcass that sinks initiates a decades-long ecological event that supports an entire universe of deep-sea organisms. Few animals on Earth contribute as much to vertical nutrient transport as sperm whales. They bridge two distant ecosystems, the productive, light-filled surface and the starving abyss far below. And they do it simply by living their lives in the only way evolution has allowed them to. The chapter concludes with the camera traveling along the length of a whale fall that has been decomposing for many years. The remaining bones glow a faint, pale color against the dark seafloor. Osedac stalks sway gently. Shrimp scuttle across sediment enriched by years of decay. In the far background, a sleeper shark drifts by, perhaps searching for remnants. This entire oasis exists because a sperm whale once hunted squid in the depths, surfaced to fertilize the ocean, and eventually gave its body back to the darkness in a final act of ecological generosity. Above, in the sunlit world, another whale prepares to dive, beginning the cycle once more. The seafloor is a place defined by patience. Unlike the surface, where sunlight fuels constant motion, the depths move according to quieter rhythms. Sediment settles in slow, steady cascades. Currents creep like wandering shadows. Most creatures wait motionless for long periods, conserving every gram of energy. But when a carcass touches the bottom, whether fish, turtle, shark, or even part of a whale, this subdued world erupts into organized chaos. The disturbance does not ring with sound the way it would on land. Instead, it radiates as scent, pressure change, and dissolved chemicals drifting outward in all directions. And the first animals to respond, often within minutes, are the crabs. The scene begins on a sandy stretch beside a coral outcrop, where the body of a large fish has just settled, belly torn from a previous predator higher in the water column. The flesh is fresh enough that its colors remain visible. Thin plumes of tissue drift upward as decomposing cells leak enzymes and lipids into the surrounding water. These signals travel far beyond the limits of sight. For crabs, whose antennae are tuned to detect even faint molecular patterns, the arrival of such a body is unmistakable. It is both opportunity and duty, the call to dismantle death before it transforms into a hazard for the reef. The first responders are usually mid-sized scavenging species, such as swimming crabs or portunids. They approach from the down-current side, following the plume of scent like firefighters tracking smoke. Their movements accelerate as the signals intensify. The crabs reach the carcass with surprising speed, scrambling over the sand in bursts of sideways momentum. Their legs probe, stabilizing the body as if inspecting its structural weaknesses. Then the feeding begins in earnest. Crabs are built for demolition. Their claws come in different shapes depending on the species. Some broad and crushing, others slender and precise. And each type serves a specific purpose in the breakdown of dead tissue. The first crabs to arrive often target the eyes and gills, not because they are grotesque trophies, but because these tissues decompose rapidly and contain soft proteins that require minimal effort to consume. Removing them early reduces the release of harmful ammonia and decreases the speed at which bacteria bloom around the carcass. In this way, crabs perform the underwater equivalent of removing the most volatile materials before deeper structural work begins. Within moments, smaller species appear. Hermit crabs scuttle over the sand, shells clinking softly as they gather around dangling tissue. They lack the crushing strength of larger crabs, so they specialize in scraping thin films of flesh that coat bones and cartilage. Their mouthparts work relentlessly, removing microbial layers, parasites, and sloughed skin. These are the detail workers, the ones who handle the finishing stages and the tight crevices others cannot reach. More species join in as minutes turn into hours. Some crabs have claws adapted to pry open joints, using serrated edges to sever ligaments that hold fins or flippers in place. Others break apart vertebrae, snapping through connective tissue that would otherwise require days of bacterial decay to weaken. Their movements are precise despite the apparent frenzy. Each claw action is calibrated, a result of millions of generations refining the ability to extract nutrients with little wasted effort. The scene might seem chaotic, but it functions like an assembly line. Each species takes what it is best adapted to handle, and in doing so accelerates the process of turning a dead animal into a series of smaller, manageable components that the entire reef can use. Unlike sharks or rays, which consume large portions of flesh, crabs rely on mechanical disassembly. They break down tissue into compact, easy-to-handle pieces, forming pellets small enough for fish, worms, or even detritus feeders to ingest. This transformation is crucial. Without crabs, dead bodies would remain intact far longer, generating stagnant zones where decomposition would consume oxygen and stress nearby corals. The camera zooms in on a single crab now, a reef-dwelling species with one claw slightly larger than the other. It positions itself near the carcass's flank and begins cutting a long strip of muscle. The claw snaps repeatedly, slicing the tissue into decreasing sizes. Eventually, the crab shapes the material into a rounded lump that it can pack between its mouthparts. Some of these lumps it eats immediately. Others are dropped on the sand, where opportunistic fish dart in to collect them. In this way, the crab creates a steady flow of processed food that ripples through the ecosystem. Fish that would never be able to tear flesh from a carcass, such as gobies, wrasses, and even shy cardinal fish, suddenly gain access to energy-rich fragments. As the camera pans outward, the entire carcass is now covered in crabs. Some hang from the ribcage, like construction workers on a scaffold. Others burrow beneath the body, searching for tender meat hidden by skeletal structures. Each crab's sensory world is a combination of chemical gradients, tactile information, and subtle vibrations. Their antennae sweep constantly, brushing across surfaces to detect softness or the presence of an opening. The clicking of claws combines with the faint rasp of scraping mouthparts, producing a soundscape that, while muted to human ears, is unmistakable to the animals that share this space. Certain crabs also play a role few viewers realize: parasite removal. Dead fish and sea turtles often host parasitic copepods, leeches or worms. As the flesh begins to break down, these parasites lose their hold and attempt to flee. Crabs snap them up immediately, preventing them from escaping into the reef where they could infect healthy animals. This role is not intentional, but evolutionary. Parasites are simply another source of protein. Yet the outcome benefits the broader ecosystem by limiting the spread of pathogens. The camera moves closer to the carcass's interior, where the organs once sat. Larger crabs have torn into the cavity, opening pathways for smaller animals to swarm inside. Fat deposits are abundant here, and crabs with large crushing claws are particularly skilled at breaking through the membranes that encase them. Fat decomposes differently than muscle. It contains long-chain lipids that bacteria are eager to consume, but that also attract specialized scavengers. Crabs speed the breakdown of these deposits, making them accessible to eels, brittle stars, and certain fish that thrive on high-energy tissue. Hours after the carcass touched the seafloor, its transformation is dramatic. Almost every visible patch of soft tissue has been removed. Bones are exposed, joints are loosened or missing. The seafloor around the body is littered with crab-made pellets, scattered flakes of processed tissue, and sediment disturbed during the feeding frenzy. This is the point at which fish arrive in greater numbers. Goatfish sift through the debris for hidden remains. Hogfish dig into the sand, uncovering pellets buried beneath flurries of sediment. Even reef sharks may return to investigate the scent left behind, though by now there is little for them to claim. Yet the crabs remain. They are the last to leave. They stay until the final scraps are gone, until the bones are clean enough for current to sweep without dislodging hidden flesh. Their persistence ensures that no nutrient is wasted. No particle large enough to rot remains untouched. When the crabs eventually disperse, they leave behind something entirely different from what they found. A clean skeleton, polished by their claws, ready to be colonized by algae and bored into by small worms. The carcass becomes a substrate instead of a hazard. One of the most important roles crabs play in this process is the conversion of unstable rot into stable food forms. Rotting tissue releases ammonia, hydrogen sulfide, and other compounds that can stress corals or smother nearby sponges. But crab-processed pellets decompose more slowly and evenly. They sink into the sand rather than floating loosely in the water. They feed detritivores without overwhelming microbial communities. In essence, crabs direct decomposition instead of allowing it to run unchecked, an ecological function as vital as the role of vultures on land. The diversity of crab species ensures that this function is always covered. On a coral reef alone, dozens of different crab species participate in carcass breakdown, each with its own specialization. Some work on the toughest parts, cracking vertebrae or limb joints. Others focus on delicate tissues like gill arches or the thin muscle sheaths that line the ribs. The variety of claw shapes, from broad crushers to slender pincers, forms a toolbox that matches every structural challenge a carcass presents. Later, when the reef quiets again and currents sweep away the last suspended particles, the sea floor shows no immediate signs of the event that just occurred. The carcass is gone, stripped to bone or completely consumed. The water is clear, oxygen levels stable, and the surrounding corals unharmed. The only lasting evidence lies in the stomachs of the animals that fed. Crabs, fish, worms, and even microorganisms nourished by the breakdown. The ecosystem has absorbed the disturbance with remarkable efficiency, largely because of the crab's relentless labor. In this way, crabs act as the bridge between death and renewal. They are not scavengers in the sloppy sense often imagined, but highly skilled recyclers whose work keeps entire reefs functioning. Their arrival marks the beginning of decomposition, but their methodical feeding prevents decay from spiraling into ecological stress. They convert danger into nourishment, rot into structure, and chaos into balance. Though they rarely receive recognition, crabs are among the ocean's most dependable engineers, ensuring that every fallen creature is transformed into life again. The chapter closes on a final scene: a lone crab walking away from the cleaned bones, pellets scattered behind it like breadcrumbs of rebirth. In its wake lies not decay but opportunity, the remains of a life now redistributed through the reef, ready to sustain the next generation of marine creatures. Night falls slowly underwater. The brightness drains from the surface layers first, turning the shallows from blue to green, then from green to charcoal grey, until only a faint shimmer trickles down through the last of the wavering kelp fronds. On the seafloor, the transition happens earlier. Rocks darken, sand cools, and crevices deepen into narrow, pitch-black tunnels. Nearly all diurnal reef life withdraws into hiding, reducing movement to a minimum in order to conserve energy until dawn. But for one group of animals, this dimming of the ocean marks the start of an entirely different shift, the march of the lobsters. The first antennal flickers emerge from beneath a boulder the size of a person. Long, sweeping feelers stretch outward like probes, sensing for danger. The lobster steps forward cautiously, its armored legs tapping the sand with deliberate rhythm. It waits, listens, tastes the current. Then more appear, another from a crevice under a ledge of coral, another from a hole carved by a long-gone sea urchin, another from beneath a shell that has become its shelter. One by one, the lobsters gather, their movements synchronized by mere chance and instinct. Under darkness, they transform from solitary territorial animals into wandering scavengers with a shared purpose: to clean the sea floor of whatever death the day has left behind. The camera follows the largest of them, an old male whose carapace is scarred from decades of territorial battles. His antennae wave through the water, detecting faint scent molecules drifting down current. These chemical signatures come from proteins breaking apart, from lipids dissolving into simple compounds, from bacteria beginning their work on a fresh carcass somewhere nearby. Lobsters are masters of this chemical world. Their antennae are covered in sensory hairs that detect concentrations so low they are invisible to nearly all other species. The scent of rot, even a few hours old, spreads through the water column in a chemical gradient the lobster reads with astonishing precision. The big male begins to walk, and the others follow the same invisible highway. Their legs move side to side, mechanical yet fluid, sending up small puffs of sediment that drift behind like dust stirred by nocturnal workers heading to a job site. These nighttime foragers do not move blindly. Every few steps they pause, test the current with their antennae, adjust their direction, and continue marching. What they track is not the fresh, intense odor that crabs respond to at the moment a carcass falls. Lobsters follow the heavier, oilier signature that develops after the crabs have already torn the first holes and exposed the deeper tissues. When the lobsters reach the carcass, the transformation is immediate. The daylight scavengers are gone. Crabs have eaten their fill and retreated into hiding. Fish have dispersed and sand clouds have settled. What remains is a partially dismantled body, ribs exposed, tendons stretched across joints like loose cables, shreds of skin clinging to muscle that still contains pockets of valuable nutrients. This is the perfect stage for lobsters, whose feeding tools are built not for fine scraping like hermit crabs, nor for surface picking like small fish, but for brute force processing. The largest lobster steps onto the carcass, gripping it with hooked walking legs. It positions itself near a joint where the crabs have already done preparatory work. Then the claws rise. Each lobster claw is an instrument of two distinct functions, one broad and heavy, meant for crushing, the other slimmer and sharper, meant for cutting. This asymmetry allows lobsters to handle brittle materials like bone and shell, as well as tough substances like sinew and cartilage. The lobster's crusher claw clamps down on the joint with force, generated through lever-like muscle attachments. The sound is not loud, but at close range the dull, cracking pop resonates through the sand. Fragments scatter. The lobster works methodically, breaking apart the structural elements the crabs could not fully dismantle. It pulls strips of tendon free, tearing them into bite-sized pieces with rhythmic claw motions. Other lobsters join in, each taking a section of the carcass and focusing on the densest parts. Vertebrae still connected by ligaments, fins with hardened rays, thick spinal tendons, and remaining organs protected by layers of muscle. This is demolition work in its purest form, and lobsters excel at it. The camera moves closer to show the mouth parts, an intricate set of mandibles and smaller appendages that shuffle food inward. Unlike many scavengers that simply bite and swallow, lobsters break their food into tiny, consistent pieces. Each chunk becomes a compact pellet as the lobster manipulates it with its mouthparts, reducing its surface area to volume ratio. This transformation is important for the larger ecosystem. Pelletized material stays in place rather than drifting away, creating stable deposits that detritivores, burrowing worms, and small fish can consume safely. Without lobsters, these leftover scraps would float across the reef, smothering coral polyps or settling into cracks where they would rot anaerobically. And it is exactly that anaerobic danger that lobsters prevent. Beneath rocks, inside crevices, and within coral labyrinths, oxygen levels can drop rapidly when decomposing tissue is left untouched. Rotting flesh produces hydrogen sulfide, methane, and other gases that stress reef organisms and kill sensitive species such as sponges and filter feeders. A carcass wedged in a narrow space becomes a biological time bomb, creating pockets of anoxia where bacterial processes accelerate uncontrollably. Lobsters, by stripping every last scrap from these tight spaces, act as natural sanitation forces that stop this process before it begins. In the scene unfolding now, one lobster crawls beneath the carcass, using its long antenna-like probes to locate any remaining tissue. It squeezes into a crevice formed by the corpse, settling unevenly on the sand, extracting pieces of meat too small or hidden for crabs to reach. Another lobster climbs the ribcage, clamping down on a stubborn tendon with its crusher claw, then pulling backward with its entire body weight to tear the last connections free. Muscle fibers stretch, shimmer, and finally snap, the equivalent of tearing reinforced cables loose from a shipwreck. The lobsters treat these tough components the same way they handle shellfish, with patience and force applied through repeated precise movements. As the lobsters continue feeding, more predators arrive quietly. Moray eels slither from dark gaps, attracted by the vibrations and scent of the activity. Instead of competing, these eels benefit directly from the lobsters' labor. The eels slip between the crustaceans, picking up fragments the lobsters drop. Eels are poor at accessing tougher tissues, but lobsters expose new pockets of meat with every single claw strike. Their cleanup opens access for a wide cast of nocturnal reef predators and scavengers, each taking a share of what the day's hunts left behind. The biological impact of lobsters extends far beyond the carcass itself. Every pellet that falls to the sand becomes a nutritional packet that fuels burrowing invertebrates. Every exposed bone contributes calcium phosphate to sediments. Every avoided pocket of anoxia allows coral colonies and sponges to continue filtering water without encountering toxic byproducts. Lobsters are not just consumers, they are stabilizers, preventing decay from becoming destructive. Hours pass, and the carcass transforms once again. What was once a fleshy mass is now a skeletal frame, dotted with fragmentary scraps. Lobsters work until even these remnants disappear. They pry apart joints that no other scavenger could handle. They break through cartilage and crack small bones into fragments the size of shells. They remove every clinging tissue, ensuring that when predators return at dawn, they find nothing left to disrupt the day's rhythm. Finally, when their antennae detect no further nutritional signals and the chemical trail fades into the ambient background of the reef, the lobsters begin their return march. The journey back is quieter. Their bodies glisten faintly under scattered moonlight that filters down in wavering beams. They retrace their steps, moving toward the crevices that shield them during daylight hours. The sand behind them shows the faintest trace of their passing. Small footprints that will vanish within minutes as currents sweep the bottom. The camera lingers on the cleared site. The skeleton rests cleanly on the sand, bleached by moonlight, empty of rot. No gases bubble from beneath it. No dense clouds of bacteria rise around it. What remains is safe, stable, and inert. A structure, not a threat. Small fish begin to pick through the area for the last microfragments. Worms emerge from the sediment to investigate. Even coral polyps extend tentacles toward the nutrient-rich particles drifting upward from the disturbed bottom. Life resumes its default rhythm. Lobsters, despite their intimidating appearance, are not apex predators or charismatic hunters. They do not chase, ambush, or overwhelm prey. Yet their ecological importance rivals that of far larger or more visible animals. They are the night shift custodians, the ones who ensure the reef does not drown in unprocessed death. Their work prevents toxic pockets from forming beneath rocks, sustains a wide variety of detritivores, and maintains the delicate balance of nutrients across the seafloor. In the darkness, when the reef is quiet and the world above sleeps, lobsters keep the ecosystem running. They are the nocturnal engineers of the ocean's recycling system, ensuring that every end of life becomes a new beginning for another, and that the night remains a place of transformation rather than decay. The kelp forest begins as a kind of underwater cathedral. Sunlight breaks through the surface in trembling beams, splintering into golden paths that drift downward until they meet a maze of swaying columns. Giant kelp rises from the seafloor like living architecture, each frond stretching toward the light with slow, deliberate grace. Schools of fish weave through the leaves, their bodies flashing silver as they turn. Crabs crawl along the base of the kelp, hiding among holdfasts that anchor the towering plants to rock. Sea cucumbers glide across the sediment, recycling organic matter into nutrients that feed the ecosystem. Everything here depends on everything else, creating one of the most productive habitats on Earth, rich in oxygen, life, and complexity. But this cathedral has a single weak point. It is built atop a foundation constantly undermined by one quiet, persistent threat: the sea urchin. The scene shifts to the forest floor, where purple and red urchins cluster around the base of a kelp stipe. Their spines click softly as they move, scraping rock with the slow determination of animals that destroy without malice. An urchin does not need to be fast. It needs only to feed continuously. Its mouth, located on its underside, consists of 5 calcium-rich plates called Aristotle's lantern, a structure evolved to grind through algal tissue, scrape rock surfaces, and slice through kelp stems. This mechanical precision turns the urchin into a significant force in the kelp ecosystem, even when its population is small. But when that population grows unchecked, the balance collapses. The collapse begins slowly at first. A few urchins gather near a base. They eat the protective tissues surrounding the holdfast, weakening the anchor. Then, in a storm or strong current, the kelp detaches and floats away. Where there was once a towering canopy, sunlight now pierces deeper, altering the microclimate of the forest floor. More urchins move into the cleared patch, attracted by the smorgasbord of broken plant material. They feed, reproduce, and expand. Their numbers grow exponentially because predators, especially their most important one, are missing. The camera widens. The kelp forest is not simply damaged, it is being erased. In places where sea otters have disappeared, the transformation is dramatic and devastating. What once thrived with dense forests becomes an urchin barren, a wasteland of exposed rock and rubble. The seafloor loses its structural complexity. Holdfasts lie in broken piles, rotting and dissolving into fragments that drift with currents. Without kelp to generate oxygen and shade, water temperatures fluctuate more dramatically. Species that once relied on the forest for food and shelter disperse or die. An entire ecological engine stalls, replaced by a monotonous plain of spiky grazers, grazing whatever tiny scraps remain. These barrens are not silent. They pulse with decay. Broken kelp, ripped loose by storms, drifts into these open zones where it settles into heaps. There, under the weight of water, the fronds rapidly decompose, releasing ammonia, methane, and other byproducts. Because kelp forests normally process detritus through complex food webs, involving isopods, amphipods, worms, and filter feeders, the sudden shift to bare rock disrupts this nutrient flow. The rotting material becomes trapped instead of recycled, smothering the seafloor. Instead of feeding life, the decomposition suffocates it. At the height of these barrens, the camera moves through a scene that feels post-apocalyptic. No towering kelp, no swirling fish schools, no shadows cast by waving fronds. Only urchins, clustered so tightly that their spines form a jagged purple carpet. Their feeding is relentless, mechanical, and utterly indifferent to the ecosystem collapsing around them. Without predation pressure, their population continues to expand, eating through every scrap of algae that dares try to grow. This is where the sea otter re-enters the story, not as a cute icon floating on its back, but as a keystone predator with ecological impact far larger than its size suggests. The transition begins with a single otter surfacing near the edge of the barren. Its whiskers glisten with seawater. Its fur, the densest of any mammal on Earth, traps air so effectively that sunlight reflects off it in a silver sheen. But the otter is not drifting lazily. It inhales deeply, arches its body, and dives. As it descends, its presence changes everything. Sea otters are obsessive in their feeding behavior. They must eat a significant portion of their body weight each day to maintain warmth in icy water. Unlike other marine mammals, they do not rely on blubber for insulation. They rely on fur and high metabolic rates. This need for constant energy drives their intense predatory nature, and their favored prey, especially when available in abundance, is the sea urchin. The camera follows the otter through the barren landscape. It moves with a sense of purpose, its front paws reaching forward with surprising dexterity. The moment it detects an urchin, it grabs the animal with both paws and ascends. At the surface, the otter flips onto its back and uses a stone, a behavior unique among marine mammals, to crack open the hard shell. The claws remove the soft gonads, the most nutritious part. It consumes them rapidly and dives again, returning to the forest floor for more. Each urchin removed has a disproportionate effect. These animals can live for decades, growing slowly but relentlessly consuming kelp. Removing even a fraction of the population allows kelp spores to settle safely and begin regrowth. But otters do not remove just a fraction. In regions where they recover, they drive urchin populations down by orders of magnitude, restoring the balance that historically shaped kelp ecosystems. The camera moves to a time-lapse sequence. Weeks pass. Where barren rock once lay exposed, juvenile kelp shoots begin to appear. Their delicate blades wave gently, almost shy in the open water. As otters continue feeding, these shoots survive long enough to anchor themselves securely. Their holdfasts grip rock, resisting currents more firmly by the day. As more kelp regrows, the shading effect reduces temperatures on the seafloor, making conditions more favorable for fish and invertebrates that avoided the barrens. Months pass. The kelp forest returns in full. Canopy layers weave together, slowing wave energy and protecting shoreline ecosystems. Fish return first: rockfish, surf perch, kelp bass, followed by invertebrates such as abalone, crabs, sea stars, and nudibranchs. The collapsing energy system is reversed. Now, momentum builds in the opposite direction, but the story does not end with visual recovery. Ecologically, the restored forest functions differently than a barren. Kelp acts as a massive carbon sink. It captures carbon dioxide through photosynthesis and locks it into biomass that can be transported offshore or sunk naturally. In a functioning forest, old kelp fronds break away gradually, sinking to deeper waters where decomposition fuels slow, stable food webs rather than chaotic local rot. The detritus becomes a steady rain of nutrients, feeding detritivores without overwhelming them. In this way, the otter becomes not simply a hunter of urchins, but a regulator of carbon flow, nutrient cycling, and structural habitat formation. By enforcing moderation on urchin populations, it prevents catastrophic release of carbon stored in kelp. It ensures that decomposition occurs at the right rate, in the right place, supporting complex detrital chains instead of triggering collapses. The camera follows an otter drifting through a forest now fully grown. It moves between the fronds with confidence, clutching a recently caught urchin in its paws. Beneath it, the holdfasts are healthy. Amphipods feed on decaying fragments. Worms burrow into sediments enriched by kelp-derived organic matter. Shellfish attach to rocks sheltered from strong currents. The ecosystem is balanced again, not static, but stable. The key concept becomes clear. The forest still experiences death. Old fronds still break free. Kelp still rots. But now, it rots in the correct context. Scattered, manageable, absorbed by the web of life instead of smothering it. The otter, through its tireless feeding, transforms the chaotic collapse of an urchin barren into the predictable pulse of a functioning kelp ecosystem. The chapter closes with the camera rising slowly through the kelp canopy. Sunlight filters through the swaying blades, casting shifting patterns on the otter floating below. The forest feels alive, vibrant, breathable again. And in the center of this restoration lies the sea otter. Soft, buoyant, and deceptively harmless looking, yet one of the ocean's most powerful regulators of life, decay, and ecological stability. The kelp forest at dawn is a living mosaic of motion and shadow. Long blades rise and fall with the pulse of the swell. Schools of perch and wrasse weave between columns of kelp, scattering fragments of light as they turn. Crabs climb through the holdfasts. The forest floor, though darker and quieter, glows faintly with life. Brittle stars tucked beneath boulders, sea cucumbers grazing along the sediment, nudie branches gliding over sponges. Among these animals sits the sea urchin, a creature whose spines cast delicate silhouettes across the sand, and whose role in this ecosystem is far more complex than its simple form suggests. At first glance, a solitary urchin looks harmless. It moves slowly, almost imperceptibly, inching over the rock with careful coordination of hundreds of tiny tube feet. Its spines sway gently as currents push past. Beneath its spherical armor lies a mouth hidden deep in the body's underside, a structure that resembles a five-pointed crown made of calcified plates, grinding and scraping with relentless consistency. This mouth, Aristotle's lantern, is one of the most effective feeding tools in the marine world. With it, an urchin can scrape algae down to bare rock, slice through kelp tissue, and chip away at coralline surfaces. But in a balanced ecosystem, this ability is not destructive, it is essential. The camera follows a single purple urchin as it grazes at the fringe of a kelp holdfast. The creature is not attacking the plant. Instead, it is trimming small algal films that accumulate around the base, films that, if left untouched, might smother the holdfast and hinder kelp growth. The urchin's grazing clears the substrate, allowing juvenile kelp spores to settle. As it scrapes, it converts thin algal layers into compact pellets that drift downward in the gentle swell. These pellets become food for detritivores, isopods, worms, microcrustaceans, feeding a cascade of small organisms that are essential to the health of the forest. At this scale, the urchin is a recycler, a janitor of the forest floor. Its feeding maintains clarity on rock surfaces, prevents the buildup of decomposing algae, and allows kelp to grow upward without competition from faster-spreading seaweeds. In this state, its presence is unobtrusive, almost beneficial. The kelp above thrives. Fish take shelter in the canopies. Sea otters patrol the forest edges. Everything is in balance. The camera shifts upward, showing the kelp canopy from above. Sunlight streams through, illuminating fronds that stretch as tall as trees in a terrestrial forest. The kelp grows rapidly. Some species can extend by more than a meter in a day, absorbing carbon dioxide as they rise, forming one of the most efficient carbon sinks on the planet. But the kelp is only as stable as the predators that regulate the animals feeding on it. If those predators disappear, the forest becomes vulnerable to a silent force waiting below. The next sequence unfolds like a slow disaster. Predation pressure drops, sea otters decline, sunflower sea stars vanish due to disease. Large fish that once preyed on juvenile urchins are fished out of nearby waters. The checks and balances falter. The population of urchins does not simply grow, it erupts. The camera returns to the forest floor. Where there was once a single urchin, now there are dozens. Their spines form thick carpets of purple and red shapes that stretch across the bottom. Their movement, though still slow, becomes collective. Instead of grazing only on algal films, they begin targeting the kelp directly. At first, they nibble at holdfast edges, but as their numbers grow, their feeding shifts from selective trimming to outright consumption. What was once a janitor is now a bulldozer. The transformation does not occur because the urchin has changed. Its behavior remains instinctual, mechanical, simple. The shift happens because the ecosystem lost its safeguards. The kelp's defensive strategies, its rapid growth, Its chemical compounds, its flexible stipes, cannot keep up with the sheer volume of mouths tearing at its base. The camera shows a kelp stalk beginning to weaken. A cluster of urchins surrounds it, grinding steadily. The holdfast fibers snap one by one, unable to resist the onslaught. Finally, the kelp detaches. It rises through the water column, carried by trapped gas in its pneumatocysts. Once anchored to the seafloor, this towering plant now drifts like a corpse. Some fronds snag on rocks. Others float toward the surface where they gather into rotting mats. These mats, once detached, no longer perform photosynthesis. Instead, they begin degrading, releasing nutrients and gases in unpredictable ways. The kelp forest canopy thins, shading decreases, water temperature rises. Herbivorous fish lose shelter, then predators lose hunting grounds. The food web destabilizes tier by tier. The urchins, sensing the absence of predators and guided only by hunger and chemical signals, expand outward in what ecologists call urchin fronts. These fronts move across the seafloor like slow-motion wildfires. The urchins advance, eat the base of each kelp they encounter, and continue forward. Behind them lies a wasteland. The camera pans across a fully formed urchin barren. The transformation is stark. Rock exposed, structure gone, complexity erased. Without dense kelp roots binding sediments, storms scour the seafloor. The nutrient cycle becomes chaotic. Decomposing kelp accumulates in pockets, creating anoxic, sulfide-rich zones where few species can survive. Bacteria bloom, amphipod populations collapse. Filter feeders starve without the steady rain of kelp detritus that a healthy forest produces. In this barren, the urchins continue feeding, but their diet shifts. With no kelp left, they consume coralline algae, microflora, and even scrape down to bare stone. Some starve slowly, yet enough survive to maintain the barren indefinitely. An urchin barren is not temporary. It is a stable alternative state, one that can persist for decades without intervention, because kelp cannot regrow with so many urchins waiting to devour any new spores. And so the urchins, once recyclers, become the architects of desolation. Yet this destructive phase reveals something deeper about their ecological role. They sit at the threshold between productivity and collapse. They are not villains, but amplifiers of whatever forces govern them. When controlled, they speed decomposition correctly, turning old kelp fronds into pellets that feed detrital webs. When uncontrolled, they accelerate decomposition incorrectly, turning living kelp into rotting debris too quickly for the forest to absorb. This duality makes them one of the most instructive animals in the marine world. The camera shifts to a surviving pocket of kelp at the barren's edge. Here, predators remain, perhaps a lone sea otter or a cluster of resilient sea stars. These predators suppress urchin numbers just enough to maintain stability. In this remnant, the urchins behave differently. They graze the rocks clean of biofilm. They trim the algae that would clog the kelp's support structures. They form neat, predictable patterns of feeding trails, their pellets falling like fine sand, feeding yet another layer of life that thrives in healthy forests. The contrast between these two zones, desolate, barren, and thriving forest, is a visual map of how predators shape ecosystems through indirect regulation. Without control, the urchin becomes a force of collapse. With control, it becomes essential to the rhythm of renewal. The chapter closes by pulling back to show the entire coastline from above. Healthy forests form dense, waving carpets of green and gold. Urchin barrens appear as pale, empty patches, scars etched into the sea floor. At first, the difference seems aesthetic, but the truth is far more profound. One side absorbs carbon dioxide, anchors biodiversity, shelters hundreds of species, and stabilizes shorelines. The other rots, releases carbon, and supports only a handful of rugged organisms. At the center of this divide is the sea urchin, an animal whose fate shapes the fate of the entire ecosystem. It is not the kelp that decides the forest's future, nor the waves, nor the nutrient load. It is the balance between urchins and those who feed on them. These spiny bulldozers reveal the ocean's simplest rule. Even the smallest recyclers left unchecked can turn living cities into deserts of decay. In the clear shallows of a coral reef, life often appears impossibly bright. Sunlight dances across corals shaped like branching trees, winding brains, sprawling plates, and delicate fans. Fish weave in and out of crevices, painting the water column with shifting colors. The entire reef hums with movement. Parrotfish scraping algae from stone, wrasses darting between coral heads, and anemones pulsing gently in the current. But beneath this surface, beauty lies a constant struggle for space, sunlight, and oxygen. Every inch of reef surface is claimed, contested, and reclaimed again. Part of this turnover is maintained by starfish. When they move, they glide as if floating on invisible tracks, propelled by hundreds of tiny tube feet arranged in neat rows under each arm. Their motion is slow but purposeful. These animals, so often dismissed as simple or passive, are in fact engineers of reef renewal. The camera moves in close to show a common reef starfish climbing over a cluster of dead mussels. Its tube feet attach and detach with hydraulic precision, gripping surfaces so delicately that the animal appears weightless. When it encounters a decaying clam, it wraps several arms around the shell, opens it with patient pressure, and everts its stomach through its mouth. Enzymes begin dissolving tissue in place, turning the carcass into a nutrient-rich slurry that the starfish absorbs. This process is essential. Without it, dead mussels and clams would accumulate in thick, smothering layers, locking up hard surfaces and preventing corals and algae from gaining footholds. Starfish transform these remains into bioavailable nutrients, clearing space for new coral fragments to settle. They help recycle weak or dying corals as well, pieces broken during storms or dislodged by fish. In this capacity, starfish are caretakers, maintaining the reef's architectural dynamism. Their role is quiet, almost invisible, but without them, reefs would clog and decay from the bottom up. The camera shifts to a time-lapse of a normal reef cycle. A coral dies naturally, its tissue sloughing away. Starfish arrive within hours, feeding on the remaining soft tissue before microbes can flourish uncontrollably. By removing dying or dead material quickly, they prevent the development of anaerobic pockets that produce sulfurous compounds harmful to the surrounding reef. They play a role similar to crabs in kelp forests or lobsters on rocky bottoms, transforming raw decay into manageable, digestible fragments. Their work keeps disease from spreading, keeps surfaces open, and keeps energy flowing through the ecosystem instead of stagnating in the bones of the reef. But the chapter does not end there, because starfish are not uniformly gentle recyclers. Some species, even within the same phylum, possess a far different ecological personality. And when environmental conditions shift in their favor, the reef's caretakers can become its conquerors. The transition begins subtly. A single crown-of-thorns starfish emerges from beneath a coral ledge. This species is unlike any of the others that share the reef. It has long, tapering arms, often more than a dozen, each lined with venomous spines. The spines glisten with toxic mucus that can cause painful wounds to divers and deter nearly all predators. Its colors are ominous, deep purples, greens, or browns that contrast sharply with the bright reef beneath it. Its stomach, when everted, releases digestive enzymes capable of dissolving the living tissue of corals within hours. One crown-of-thorns starfish is manageable. A few are normal. They are part of the reef's natural balance, feeding on fast-growing coral species and preventing any one type from dominating. But when predators decline, especially large fish like Napoleon wrasser, giant triton snails, or other specialized starfish eaters, the balance shifts. Reduced predation, combined with nutrient spikes from land runoff or changes in ocean conditions, leads to an explosion in juvenile starfish survival. What begins with dozens can multiply into hundreds, then thousands. This is when the reef faces one of its most terrifying transformations. The camera pulls back to reveal an outbreak in progress. Crown-of-thorns starfish cover the reef like a moving carpet of spiked bodies. They advance together, each animal extending its stomach onto coral heads, dissolving tissue with chemical precision. What remains behind them is unmistakable: a white, skeletal framework stripped of life. Within days, algae begins to colonize the exposed surfaces. Within weeks, the algae thicken, trapping sediments and suffocating any surviving coral fragments. What was once a kaleidoscope of color becomes a graveyard. The destruction is not random. Crown-of-thorns starfish exhibit coordinated directional feeding patterns known as fronts. These fronts move across the reef like slow-motion wildfires, consuming everything in their path. Unlike urchin barrens, which often arise gradually through overgrazing, crown-of-thorns outbreaks strike with rapid intensity. The starfish digest living coral tissue, not dead or dying tissue. They feed on the reef's architects, stripping the ecosystem down to bare skeleton. The next sequence shows a coral reef before and after an outbreak. Before, branching corals teem with fish, forming complex homes and feeding grounds. After, the fish vanish. The corals collapse. The skeletons lose structural integrity as algae, sponges, and microbes infiltrate the porous framework. Decay sets in rapidly, leaving mats of rotting biofilm that attract detritivores but offer no stable habitat. The reef becomes flat, uniform, and nearly devoid of shelter, a biological desert. The dramatic contrast illustrates a powerful ecological truth. Starfish regulate the reef's stability not by existing, but by how many exist. The camera turns to the few predators capable of controlling crown-of-thorns populations. Chief among them is the Napoleon wrasser, a massive, thick-lipped fish capable of crushing the starfish's spiny arms with its jaws. Another is the giant triton snail, whose venomous proboscis can immobilize and consume even fully grown starfish, but both predators are vulnerable to fishing pressure. When they disappear, the reef loses its top-down control mechanism. Juvenile crown-of-thorns starfish, which normally suffer high mortality under predation, survive in overwhelming numbers. Their larvae drift on currents far beyond the boundaries of any one reef, establishing new outbreaks across entire regions. The film zooms in on the starfish's digestive process. When feeding, it envelopes the coral with its stomach, dissolving tissue through external digestion. This method gives it access to nutrients locked inside corals without needing to bite or tear. It is a form of chemical predation, efficient and devastating. Once the tissue is dissolved, the skeleton is left behind, vulnerable to erosion and bacterial decay. Over time, the skeleton crumbles, removing physical structure essential to fish nurseries and invertebrate habitats. The next moment reveals a sinister feedback loop. As starfish destroy coral, the reef becomes more vulnerable to storms, bleaching events, and disease. With fewer predators, the next generation of crown-of-thorns survives in even greater numbers. Nutrient runoff from land fertilizes the plankton that their larvae feed on. Warmer waters accelerate their development. A single outbreak begets another, and another, until entire regions collapse into algal plains. But the chapter also shows the other side, the recovery. The camera follows a healthy reef where predators remain abundant. A Napoleon wrasser patrols the coral heads, crushing young starfish before they grow large enough to reproduce. A giant triton snail glides across the substrate, leaving a trail of mucus. When it encounters a crown-of-thorns juvenile, it consumes it efficiently. On this reef, starfish densities remain low. They fulfill their natural role as recyclers, consuming weak coral tissue, trimming fast-growing species, recycling decayed shells, and maintaining ecological diversity. The reef remains vibrant, shimmering with the full palette of corals and fish. The closing scene zooms out once more across both worlds, the thriving reef and the dead one. The difference between them rests on something deceptively simple. Whether starfish remain caretakers or become tyrants, their biology does not change, their instincts do not shift. It is the balance of predation, nutrient cycles, and environmental stability that determines their role. Controlling starfish outbreaks is not about removing them entirely. It is about restoring the forces that keep them in check. The predators, the water quality, the equilibrium that turns them from destroyers back into recyclers. In this way, starfish reveal a profound paradox of reef ecology. The creatures that clean and rebalance the system when populations are stable are the same creatures that dismantle and rot it alive when stability falters. They are the reef's quiet custodians and its crowned tyrants, depending entirely on whether the ecosystem around them holds firm or slips into imbalance. The first light of morning spreads across the reef in faint gold ribbons that flicker through the water like drifting silk. Parrotfish clatter against corals, surgeonfish shimmer in tight formations, and wrasses dart in rapid bursts through branching shelters. The reef stirs awake with its usual chatter of scraping, sifting, and feeding, but one presence moves through the awakening city with far more gravity than the rest. A single, massive Napoleon fish. It appears slowly from behind a coral buttress, its enormous forehead ridge shifting in the refracted light. Its scales shimmer in blues and greens so deep they seem carved from the reef's own palette. Thick lips and a powerful jaw form a silhouette unmistakable to any predator or prey that encounters it. The Napoleon fish, also known as the humphead wrasser, is not merely a large fish. It is an apex enforcer, a living counterweight that holds entire sections of the reef together by deciding what lives long enough to feed and what dies before it spreads rot. The camera glides closer to reveal the fish's true weaponry. Its jaws are lined with teeth, not meant for slicing, but for crushing. Behind these visible teeth lies a second set of pharyngeal teeth, a reinforced grinding mill capable of pulverizing shells, spines, and toxic tissues that would defeat almost any other reef predator. The fish surveys the reef with slow, deliberate movements, its powerful fins steering it with minimal effort. It is not searching for the same prey that smaller wrasses chase. It is searching for harder targets, targets that most of the reef's population cannot touch. A crown-of-thorns starfish emerges from behind a coral head, moving across the reef like a spined nightmare. The starfish stretches more than a dozen arms outward, each armed with venomous spikes coated in toxins strong enough to cause intense pain and swelling in large mammals. For most fish, a crown-of-thorns starfish is an untouchable, chemically armored threat that consumes coral uncontested, but not here, not under the gaze of the Napoleon fish. The giant wrasser approaches slowly, pivoting its body to face the starfish directly. The starfish senses vibrations and attempts to retreat, drawing its arms inward. It is too late. With a sudden thrust, the Napoleon fish lunges forward and clamps down on one of the starfish's arms. The sound is muted underwater, but the crushing power is unmistakable. The spines snap, toxic tissue ruptures. The starfish contorts, attempting to detach, but the fish bites again, crushing a second arm. The predator lifts its head slightly, shakes its powerful jaws, and ingests the mangled limb. To the Napoleon fish, the venom is irrelevant. Its stomach lining, mucus layers, and detoxification enzymes are evolved to withstand what would disable or kill many other species. This is what makes the Napoleon fish essential in preventing runaway decay on coral reefs. Crown-of-thorns starfish, when uncontrolled, dissolve living coral tissue at terrifying speed. They leave skeletons exposed to algal overgrowth, bacterial mats, and rot. But a single large Napoleon fish can consume dozens of juveniles and young adults in a week, intercepting the outbreak before it becomes catastrophic. It is one of the only predators strong enough, bold enough, and tough enough to bite directly through the starfish's spines. The camera follows the Napoleon fish as it resumes patrolling, moving with unhurried confidence. Its next target reveals another side of its ecological importance. A sea cucumber lies on the reef floor, expelling a thin line of sticky defensive threads into the water. These threads are laced with toxins designed to immobilize or choke small predators. Sea cucumbers are vital recyclers, but when populations explode, they can smother sediment with undigested detritus, altering oxygen flow and reducing habitat quality for filter feeders and small invertebrates. Most predators avoid them. The toxins alone are enough to discourage almost any fish. But the Napoleon fish glides above the cucumber, tilts slightly downward, and delivers a decisive bite. The cucumber folds in half as its body wall ruptures. The fish shakes the prey once, twice, and swallows a large portion of it, ingesting toxins that would sicken most fish species. The Napoleon fish then crushes the remaining half of the cucumber with slow, grinding motions of its pharyngeal teeth. Nothing remains but small fragments scattered on the sand. These pieces now feed scavengers that would never dare approach the intact cucumber. In this moment, the Napoleon fish reveals its role as a regulator of the reef's recycling process. It does not prevent decay, it controls the tempo of decay. It ensures that decomposers, grazers, and filter feeders do not accumulate in numbers that destabilize the reef's nutrient flow. It suppresses the species capable of turning the reef's delicate recycling rhythm into a runaway collapse. The camera approaches a rocky crevice where several urchins huddle. These urchins are smaller and less armored than the massive purple urchins of colder kelp forests. But their effect is similar. Unchecked, they can graze coral bases, scrape algal films too aggressively, and destabilize the coral's ability to compete with faster-growing algae. Sea urchins are essential recyclers, but only in moderation. The Napoleon fish dives toward the crevice. It uses its thick lips, designed like padded tools, to reach between sharp rocks without injury. It clamps down on an urchin and lifts it free. Spines bend, crack, and snap as the fish exerts pressure. In a few seconds, the urchin collapses into fragments. The fish swallows the soft interior and allows the shell remains to scatter. These crushed fragments now serve as calcium sources for coralline algae and microinvertebrates, feeding a miniature cycle of growth that would not occur without the predator's intervention. In this single behavior, the Napoleon fish reveals another subtle truth. It is not merely preventing overgrazers from stripping the reef, it is transforming dangerous prey into beneficial resources. The chapter widens to show how this predator's presence influences entire regions. In areas where large Napoleon fish persist, coral cover remains higher. Outbreaks of crown-of-thorns starfish stay localized or never emerge at all. Urchin numbers stay balanced. Sea cucumber populations remain diverse instead of monocultural. Algal mats do not smother coral skeletons. Decomposition remains a controlled pulse rather than a suffocating surge. But where Napoleon fish disappear, from overfishing, habitat destruction, or targeted trophy removal, decay accelerates. Crown-of-thorns populations soar, turning vibrant coral landscapes into wastelands of bright white skeletons. Urchins proliferate, grazing down juvenile corals and clearing space for algae to dominate. Sea cucumber explosions blanket the sediment with thick layers of processed organic material that reduce oxygen flow. The reef shifts into a state where rot does not wait for natural cycles. It runs ahead unchecked. The Napoleon fish is one of the few predators that bites directly into the core of this problem. Literally, its jaws enforce stability. Its appetite suppresses ecological collapse. Its presence keeps the reef's recycling system running at a controlled speed rather than burning out of control. The camera returns now to the fish itself, cruising through the central corridor of a healthy reef. Sunlight highlights the edges of its fins. Small, cleaner wrasses swim alongside, removing parasites from its scales. The Napoleon fish pauses briefly near a coral head, sensing the faint chemical signature of another crown-of-thorns juvenile hiding in a shaded pocket. Its body tenses slightly, then it lunges, biting down on the hidden starfish in one decisive motion. The reef, as always, remains unaware of the catastrophe just averted. The final sequence pulls away into a wide aerial shot of the reef. Coral heads glow with vibrant color. Fish move through branching shelters. Sponges filter water. Everywhere, tiny fragments of decay are processed in the right order. Old tissue dissolves. Detritivores feed. Algae settles in patterns the corals can tolerate. Grazers trim it back. Corals grow anew. And at the edge of this vast, intricate machine swims the Napoleon fish, the anti-rot paladin, the armored guardian who ensures that decomposition fuels the reef's life cycle instead of overwhelming it. Its jaws, more than any blade or tooth in this ecosystem, keep decay from becoming destruction. Open water always appears deceptively empty. The sunlight penetrating the surface seems to illuminate nothing at first, just shafts of blue radiance descending into depth, dancing across faint ripples in the water column. But wait long enough, and shapes begin to emerge from the monotony. A distant school of fish drifts past like a single organism. A drifting jelly pulses lazily. A lone shadow cuts across the sun's reflection for a fraction of a second. This space, which seems barren from above, is in truth a biological arena where the slightest weakness becomes visible, and where predators act long before observers could guess what is happening. The camera glides through this bright pelagic world until movement catches the lens. A long, sleek form, silver as a blade, and poised with almost unnerving stillness. The great barracuda holds position just below the surface, suspended by micro-adjustments of its fins. It faces slightly upward, reflecting sunlight along its flanks in a flickering pattern that breaks its outline. Its body is elongated, built like a living spear. Its jaws, partially open, reveal a row of interlocking teeth so sharp and widely spaced that even water seems to hesitate around them. Nothing about this predator looks uncertain. It is a specialist built for decisive action. The barracuda's eyes remain steady, scanning the water ahead. To us, the view seems empty, but to the fish, the column is filled with information. Pressure waves from distant kicks, polarized light patterns revealing silver scales, faint chemical cues drifting from stressed or wounded animals. A school of fusiliers appears in the distance, a shimmering, coordinated group moving in flawless synchrony. Their formation is tight, controlled, and constantly shifting. To a skilled hunter, the school's perfection is not what captures attention. It is the imperfections. The camera zooms in on the school. Near the tail end of the formation, one fish lags slightly behind, its movements a fraction of a second out of sync. The difference is nearly invisible, but in the pelagic environment, that fraction may as well be a scream. The fish's scales reflect oddly uneven patterns, signs of parasite clusters lodged between them. A small injury on its flank alters the fluid dynamics of its swimming stroke. Its gill plates flutter harder as it struggles to maintain oxygen flow. To us, it is nearly identical to the others. To a barracuda, it is a beacon. The predator shifts. Its dorsal fin rises slightly, a silent readiness signal. The barracuda no longer floats. It calibrates. Muscles coil beneath its shimmering skin. The slightest bend of the body realigns all its aerodynamic forces. Its motion is not a chase, it is a measurement. Barracudas hunt by reading silhouettes. Healthy fish maintain tight, predictable shapes when viewed head-on. Their bodies move rhythmically, forming a continuous visual wave when grouped. Sick fish break this wave. Their bodies wobble, their tails drag. They make irregular micro-corrections. These distortions stand out like neon warnings against the uniform backdrop of the school. A barracuda sees these flaws with almost surgical precision. The strike happens in less than a second. The camera barely tracks it. One moment the barracuda is a suspended silver rod. The next, it is a bolt of pure speed. Its acceleration is astonishing. A burst fueled by explosive muscle fibers and a body built to minimize drag. The predator does not waste time correcting course. It commits instantly. Its jaws open wide, forming a V-shaped silhouette with gaps that allow water to pass through rather than pushing against it. This anatomical trick lets the barracuda bite at full speed without slowing, a rare evolutionary achievement. The jaws close around the trailing fusilier. The bite is not a simple clamp. Those iconic fang-like teeth shear through muscle and bone with ease, slicing the fish nearly in half. The barracuda swallows the stunned prey in seconds, then glides forward again, resuming its vigil with the same eerie stillness it displayed before the strike. To the casual viewer, this may appear like mere opportunistic predation, a hunter picking off the weak because they're easier targets. But from the reef's perspective, this act is far more consequential. Barracudas perform a filter function for open water ecosystems. By removing sick, injured, and parasite-loaded individuals, they prevent diseases from spreading through entire shoals. The camera reveals the school in the aftermath of the strike. They tighten their formation, but their movement remains fluid. By eliminating the lagging fish, the predator has restored uniformity to the group. Unhealthy individuals often carry pathogens that can sweep through densely packed fish communities. Parasites that lodge in gills and spread through direct contact. Bacterial infections that bloom in micro wounds, or viral agents that take advantage of social clustering. If left unchecked, these diseases can cascade upward through the food chain, infecting predators that consume large numbers of contaminated prey. The barracuda's predation is therefore not simply personal survival, it is population-level quality control. The scene shifts deeper, where the water grows darker and the light takes on a faded green hue. Here, another barracuda patrols between shifting curtains of plankton. Below it drifts a loose aggregation of herring. These smaller fish rely on density as their primary defense. They twist and turn in hypnotic waves, confusing predators with unpredictable flashes of silver. But when one fish weakens, it loses its ability to maintain perfect alignment. Its reflexes slow, its tail beat becomes erratic. In a world where timing and synchronization mean survival, even the slightest lapse is fatal. This herring, infected by a parasitic copepod lodged inside its gill cavity, struggles to keep pace. The copepod steals oxygen and nutrients, making the fish gasp harder. Gaps appear in its swimming rhythm. Its body falls half a beat behind. This half beat is fatal. The barracuda sees it instantly. The predator does not need to attack the center of the school. It knows the importance of minimal effort and maximal reward. Just as before, it launches with a precision that borders on mechanical perfection. Its body flattens into a streamlined shape, minimizing turbulence. It strikes not where the fish is, but where it will be after half a second of delayed reaction. The jaws snap shut. The copepod-infested herring disappears. Disease that could have traveled to dozens of individuals ends here, absorbed into the stomach of a predator, built to neutralize biological threats before they spread. The chapter then shifts to a surprising angle. Barracudas do not only detect sick individuals visually, they also track subtle chemical cues released by stressed or dying fish. A fish with failing immune defenses releases a different chemical signature into the water. Changes in mucus composition, altered hormone levels, and increased ammonia output. A barracuda cruising upcurrent can detect these tiny deviations and follow them to their source. This chemical hunting strategy allows them to spot illness long before it manifests visibly. The camera then shows a nighttime scene. Under a darker sky, the water becomes quieter. The barracuda's reflective scales blend into the moonlit surface. Now it hunts fish attracted to the top by plankton blooms. Fish often weakened by parasites that alter their behavior. Many parasites manipulate hosts to make them more visible or accessible to predators, completing their life cycle only when the host is consumed. In these cases, the barracuda is both predator and unwilling participant in parasite evolution, taking out individuals whose altered behavior could jeopardize the shoal. Over thousands of generations, barracuda pressure has shaped fish populations into tighter, healthier, more coordinated schools. Their presence forces prey to maintain peak performance. Weak genes diminish, strong behaviors persist. The barracuda is not a sculptor of bodies, but a sculptor of thresholds. It sharpens the line between survival and collapse. The final scene returns to open water at midday. A school of sardines swirls like liquid metal. A single barracuda shadows them from afar, not attacking, simply watching. The school's formation is perfect. No weak stragglers, no erratic swimmers. For now, the sniper has nothing to shoot. Its role is passive, yet profound. It enforces order by simply existing, and the system knows it. The ocean stays clean not because rot never appears, but because predators like the barracuda remove rot before it spreads, before it poisons, before it reshapes the water column into a breeding ground of sickness. It removes those whose muscles fail first, whose immune systems falter, whose gills lose rhythm. In doing so, it protects not only the school, but the predators that feed on the school, and the predators above them, all the way up the ladder. The barracuda is not the villain of open water. It is the sniper that prevents biological collapse. It is the surgeon that operates without tools. It is the silent regulator of pelagic life, ensuring that every breath taken by every fish is part of a system that never allows rot to outrun resilience. The open ocean widens beneath the morning sun, a blue desert shaped by currents and illuminated by shifting curtains of light. To the untrained eye, it appears empty, but beneath the surface, life is gathering. A school of baitfish stretches across the water column like a glimmering tapestry. Thousands of individuals moving as one, each body reflecting sunlight in tight bursts of silver. The school pulses with impeccable discipline, every fish maintaining the same distance from its neighbors, the same rhythm, the same synchronized turns. From afar, they form a single organism, coordinated, cohesive, and seemingly impenetrable. But the ocean is never impressed by unity. It tests it. A faint series of clicks echoes through the water. At first, they sound like scattered pulses of static, but within seconds they sharpen into a focused pattern. Dolphins are approaching. Their bodies appear as curved silhouettes slicing through the blue, each movement smooth and measured, engineered by evolution to maximize speed, agility, and precision. A pod of dolphins does not arrive quietly. They announce themselves through waves of sound far more complex than any predator's roar. Echolocation pulses travel outward in tight beams, bouncing off scales, bones, and organs. In a single burst of sound, a dolphin reads the school, detecting density, direction, and even the individual health of fish. The baitfish tighten their formation. Something ancient and instinctive tells them the test has begun. The dolphins accelerate. Their bodies, lined with muscles optimized for sudden bursts of power, create narrow trails of pressure that ripple behind them. They do not strike immediately. Instead, the entire pod circles the school, carving a fluid ring around the swirling mass of fish. This behavior is not just hunting, it is assessment. The dolphins gather on the perimeter, swimming in loops that compress the school tighter with every pass. The fish respond by drawing inward, forming a dense ball that shifts and warps like a living planet. The center grows almost solid, while the outer shell struggles to maintain cohesion. The pod begins its first test. A dolphin charges the fishball at high speed, pulling up at the last second. Its body passes within centimeters of the outermost layer, sending a shockwave through the formation. The fish react instantly, snapping into a synchronized ripple that travels from one side of the ball to the other. Most individuals respond flawlessly, turning in perfect coordination, maintaining equal spacing. A smaller group reacts a fraction of a second too late. To a dolphin, that fraction is the entire story. Dolphins hunt not just to feed, but to probe. They use these high-speed passes to force the school into repeated evasive maneuvers, draining energy from fish already struggling to keep pace. This is selection in motion. Every feint, ram, and sudden slash through the school is not a random attack, but a diagnostic tool. Healthy individuals maintain pace. Weakened ones falter. Injured fish peel off under the strain, their muscles burning faster, their oxygen depletion accelerating. Those harboring parasites lose coordination sooner. Those with infections or micro-injuries show their weakness under pressure. The dolphins read these failures with astonishing clarity. The next moment, the pod shifts strategies. Instead of circling as a group, the dolphins begin tag-team assaults, diving beneath the school and erupting upward in explosive bursts. Their jaws snap shut on empty water or clusters of fish, but the goal is not yet to kill. The goal is to break shape. Each attack forces the school to compress, expand, twist, and reassemble. These constant contractions and expansions expose subtle differences in stamina. Some fish struggle to maintain the rapid changes in direction. Their bodies lag. Their tail beats lose evenness. These outliers do not just risk death, they invite it. The pod now coordinates more tightly, funneling energy like a shared mind. One dolphin swims above the school, casting a shadow that forces the fish downward. Another approaches from the rear, driving the school forward. A third rushes through the side, attempting to split the mass. The pattern is relentless, exhausting. For the fish, this is a trial by fire. Only the strongest remain perfectly synchronized. Only those with robust cardiovascular systems maintain the pace. Only those free of parasites, disease, or age-related degradation can keep up with the school's collective pulse. A single fish, exhausted by the barrage, drifts outward by mere centimeters. Its tailbeat rhythm breaks for only a moment. That moment is enough. A dolphin angles upward with a slight twist of its body. Its speed increases. Its mouth opens. The fish is captured in a flash of white foam and broken scales. The rest of the school tightens instantly, regaining perfect formation. The removal of this one weak link strengthens the entire group. The pod continues its assaults, each pass exposing another candidate for removal. This is the dolphin's role in the ocean, pressure that sculpts, purifies, and refines. The camera shifts to a deeper, bluer region where sunlight fades into dim turquoise. Here, the test intensifies. Dolphins herd the school downward, forcing fish to swim against buoyancy. This vertical pressure drains energy faster. Fish, gasping for oxygen, separate first. Those with compromised gills, those carrying internal parasites, those whose immune systems struggle under stress. The dolphins exhibit remarkable restraint. They could kill at any moment, but they prefer to hunt efficiently with minimal energy expenditure. Weak fish separate themselves, peeling off automatically, surrendering to the predators. The pod now switches from assessment to execution. A dolphin charges the outliers with a burst of speed, its jaws clamp shut with a crunch. Another dolphin targets a different straggler, catching it mid-turn. The pod consumes several fish in quick succession, each bite removing biological liabilities from the ecosystem. These prey are not typical. They are fish that would have died slowly from disease, spreading pathogens through the school, weakening the overall population and attracting opportunistic predators that destabilize the food web. By removing them, dolphins perform an invisible service. They prevent rot before it begins. The camera returns to the main school, which is now reformed perfectly. The fish swim tighter, faster, and more coordinated. They have learned new escape maneuvers, refined their responses, strengthened their bonds. They will survive future predators more effectively because of this encounter. In the ocean, evolution is not just shaped by who dies. It is shaped by who is pressured to survive. But dolphins do not test only for sickness. They also test for reaction speed, endurance, maneuverability, and cohesion. They push the school until the weakest reveal themselves. Those who remain become fitter, more resilient, more capable of sustaining the population. This selection pressure radiates outward through the entire oceanic system. Healthy fish populations support healthier predators. Healthy predators keep prey populations regulated. Disease outbreaks diminish. Energy transfer becomes more efficient. Entire ecosystems benefit from a predator willing to hunt in ways that appear unnecessarily elaborate to human observers, but that are, in truth, essential. In another sequence, the camera shows a pod teaching its juveniles. Young dolphins mimic the adults' movements, testing schools of smaller fish. Their attempts are clumsy at first. Underestimated angles, mistimed bursts, missed targets, but the learning is immediate. The young dolphins gain experience by repeating the same pressure routines, circling, feinting, chasing, and reading the subtle signs of weakness in prey. Through practice, they become expert selectors, understanding the delicate balance between culling and preserving, between exerting pressure and allowing strength to grow. This inherited knowledge allows dolphins to maintain their ecological role across generations. They become the constant force, ensuring that fish populations do not slide into weakness or disease-driven collapse. The ocean's clarity, diversity, and stability rely on predators that do far more than kill. Predators that test, shape, and refine. The chapter ends as the sun sets across the open water. The school drifts into safer currents, tighter, stronger, unified by survival. The dolphins disperse, diving into deeper waters with smooth, effortless strokes. Their work is done. For now, they will continue to hunt, test, probe, and challenge. They will continue to sort the healthy from the sick, the capable from the compromised. In the vastness of the ocean, where decay can spread silently and sickness can ripple through populations unnoticed, dolphins act as sentinels. Their intelligence is not just behavioral. It is ecological. Their hunting is not just predation. It is regulation. Their pressure is not destruction. It is renewal. They do not simply eat the weak. They shape the strong. Along the world's coastlines, where waves break into sheets of white foam and tides sculpt the boundary between land and sea, an overlooked engine of marine productivity pulses quietly beneath the surface. Rocky shores slam against the open ocean. Ice edges crack with the rhythms of distant swells, and every rising tide brings with it a new cargo of drifting bodies. Dead fish, storm-killed penguins, kelp torn from reefs, fragments of whale flesh, and the shredded remains of creatures lost to deeper predators, all wash ashore in chaotic waves. These coasts are not just boundaries. They are collection zones, graveyards where the ocean's debris comes to rest. The camera pulls close to one such shoreline, a jagged volcanic coast in the subpolar zone. Foam crashes violently against black rock. Between tides, debris gathers in pockets where gulls circle and crabs crawl, but the dominant presence here is unmistakable. Seals. Their silhouettes appear first as dark, round shapes lounging across boulders, but once the tide turns, they become mobile, curious, and opportunistic. Unlike their depictions as sleek hunters chasing fish in clear water, seals spend much of their time working the edges, scavenging whatever the sea offers up. A solitary seal lifts its head. Its whiskers, thick and sensitive, twitch as it picks up the faint chemical trace of a drifting carcass. Seals possess exceptional olfactory sensitivity, especially when scanning air currents at the surface. The animal slides down the rock with surprising grace and moves toward the waterline, weaving between spray-wetted stones. It times its descent with the swell, waiting for a wave to pull back, then lunging forward as the next crest lifts a battered fish carcass onto the rocks. With a quick bite, the seal claims the offering, dragging it higher before the surf can reclaim it. This scene unfolds repeatedly along rocky coasts, seals scavenging. They take what gravity and hydrodynamic turbulence deliver, and their timing is impeccable. Unlike purely pelagic hunters, seals know the value of the tide line. The ocean's currents concentrate organic matter there, forming a reliable buffet that requires far less energy than deep dives or high-speed pursuits. For the seals, each carcass is more than a meal. It is a link in a nutrient chain that stretches from the shoreline to offshore food webs. But the story widens. The camera pans across a colony. Dozens of seals haul themselves onto ledges, their bodies glistening with seawater. Some rest, shedding skin in small flakes that drift into tide pools. Others defecate into the surf, releasing nutrient-rich wastes that dissolve quickly into the churning water. These skin fragments, fecal plumes, and leftover scraps of scavenged carcasses all contribute to a subtle but powerful chemical signature that transforms the coastal zone into a nutrient pulse generator. At high tide, waves swirl through these colonies, picking up the organic particles that seals leave behind. Feces rich in nitrogen and phosphorus dissolve rapidly, while softer tissue remnants from carcasses break apart into fine particulate matter. As tides pull back, these particles swirl outward into the nearshore waters, a cloud of nutrient-rich soup invisible to the naked eye, but biologically explosive once it mixes with sunlight and coastal currents. The camera transitions underwater, revealing the transformation. What appears as normal seawater becomes increasingly turbid with microscopic life. Phytoplankton, the engine of the marine food web, bloom in response to the sudden nutrient influx. These single-celled organisms divide rapidly, turning the once clear water into a faint green haze. The change is subtle but significant. A bloom seeded not by distant upwelling or storm-driven mixing, but by the steady release of nutrients from a seal colony that doubles as a coastal composting station. Plankton blooms feed zooplankton, copepods, krill, larval fish, which trace these chemical gradients with astonishing sensitivity. Soon, clouds of tiny organisms spread across the nearshore zone, turning it into a feeding ground for small fishes that graze on plankton like herds moving through underwater pastureland. These plankton-eating fish multiply rapidly, forming schools dense enough to attract pelagic hunters, mackerel, herring and sardines, which arrive in shimmering waves. And then the seals, after resting on shore and digesting their earlier scavenged meals, push back into the water to hunt these very fish. The camera follows a seal diving beneath a baitfish school. Its body moves with surprising agility, folding and twisting as it targets the dense clusters of fish that fed off plankton grown from nutrients it released earlier. Every movement is efficient. A rapid spin, a burst of acceleration, a sharp turn that drives prey toward the surface. The seal captures one, then another. Its powerful jaws snap shut, and it swallows the fish whole. The cycle continues. Seals feed, seals return to shore. Seals deposit nutrients. Nutrients feed plankton, plankton feed fish, fish feed seals. What appears chaotic is, in fact, a feedback loop. A biological engine in which decay fuels life and scavenging powers productivity. But the ecological role of seals extends deeper than this single loop. On icy coastlines, where penguin colonies huddle between glaciers and sea ice fractures into fields of blue shards, seals patrol the ice edge for different kinds of offerings. Dead penguins caught in storms, fish frozen in surface ice, chunks of whale blubber gnawed by orcas, krill-laden slush, all drift into seal territory. Ice edges act as conveyor belts for biological debris, and seals are among the first to intercept the cargo. A Weddell seal moves into frame, gliding beneath a raft of ice. Its whiskers detect the faint turbulence of sinking organic matter. With a subtle twist of its body, it rises toward a drifting penguin carcass, trapped against the underside of the ice. The seal bites into the soft tissues, consuming the nutrient-dense sections, while leaving bone fragments and feathers to drift downward. On the seafloor below, these remnants become food for scavenging amphipods and benthic invertebrates, creatures that in turn fertilize sediments and feed filter-feeding species that line the shallow continental shelf. In this icy environment, seal waste behaves differently. Fecal plumes sink farther, carrying nitrogen and iron deep into the water column. These nutrients seed under ice algal growth, which forms green films along the ice underside. Krill graze these films, expanding rapidly in number. Whales feed on the krill. Seals feed on the whales' leftovers when carcasses wash ashore months later. Every piece of this system loops back to the coast. To understand how coastal nutrient cycles evolve, the camera zooms into a microscopic world. A droplet of water taken from the surf zone reveals a galaxy of activity. Diatoms with silica shells drifting in spirals, dinoflagellates spinning like cellular gyroscopes, bacteria clustering around organic particles shed by seals and carcasses. The bacteria break down these particles, releasing more nutrients into the water. This microbial decomposition is the foundation of the bloom, a process accelerated dramatically by the presence of seal colonies. The chapter shifts to a quieter moment on a rocky beach. The tide has pulled out, leaving pools teeming with snails, worms, and juvenile fish. Seals lie sprawled across the rocks, their bodies warm under the sun. Some shed skin in small translucent patches. These skin fragments fall into crevices. When the tide returns, the fragments dissolve into the surf. This gentle shedding contributes a surprisingly consistent flow of organic matter, not unlike leaf litter in a forest, that feeds microbial communities and supports a chain of nutrient recycling. The seals eventually return to the water, propelled by hunger or instinct, their bodies slipping between waves with practiced ease. What they leave behind continues feeding the system until they return again. Their presence becomes a biological tide in itself, synchronized with the ocean's rhythm, but governed by the ecology of the shoreline. Near the end of the chapter, the camera rises high above the coastline, revealing the scale of the feedback loop from a bird's-eye view. Plumes of nutrient-rich water extend outward from seal colonies like ghostly fingers, invisible but ecologically potent. These plumes drift offshore, creating pockets of high productivity that attract fish schools in vast numbers. The fish attract larger predators. Seals hunt the returning fish. The cycle closes. Rotting matter, whether from waves or from the seals themselves, is not a dead end. It is the ignition point for a system that transforms decay into abundance. The chapter closes with a serene underwater view beneath a blossoming phytoplankton cloud. Tiny green cells swirl like dust in sunbeams. Juvenile fish dart through the haze, feeding ravenously. In the distance, a seal glides through the water, sleek and calm, its body silhouetted against the shimmering cloud of life it helped create. Here at the coastal edge of the world, decay is not failure. It is momentum. It is a seed. And seals, scavenging quietly along rocky shores and shifting ice, are the gardeners of that fertile rot, turning graveyards into food factories and death into the engine of coastal renewal. The boulder field lies in the twilight zone of the shallow reef, where light still reaches but arrives weakened and fractured, filtered through drifting plankton and shifting curtains of sand. The rocks are worn smooth by centuries of tidal motion and storms, their surfaces mottled with patches of encrusting algae and tiny barnacles. At first glance, the place feels quiet, almost barren. But a closer look reveals subtle signs of activity. A crab leg wedged between two stones, a snail shell with its operculum missing, a pile of clam valves fanned out like discarded pages of a book. These scattered remains tell a precise story. Something lives here that eats armored prey for a living. The camera drifts toward a cluster of boulders. There, beneath a ledge, sits the unmistakable entrance to an octopus den. The entrance looks like a miniature quarry, a ring of broken shells and crab bits forming a jagged halo around the opening. This is not accidental. Octopuses do not tidy their homes. They maintain them by pushing the remains of every meal outward, clearing space inside for safety, eggs, or rest. The result is a distinctive midden, a circular heap of death that advertises the predator's presence yet simultaneously protects it by forming a spiked, textured perimeter that discourages other animals from crawling too close. Inside the den, two bright eyes watch the world with unsettling intensity. The octopus sits low, its skin shifting patterns in slow pulses, mottled brown one moment, stone grey the next. Chromatophores expand and contract beneath its surface, allowing rapid camouflage against the boulders. Its arms coil lightly, not in tension but in readiness, each equipped with dozens of suckers capable of tasting, gripping, and analyzing anything they touch. For an octopus, arms are not tools. They are minds of their own, each capable of independent problem-solving. A small crab scuttles across the sand, unaware of the danger only meters away. The octopus senses it through vibration, an almost imperceptible tremor traveling across the substrate. Its pupils narrow, its arms slide outward with fluid control. There is no dramatic rush. The octopus simply extends, melts forward, and engulfs the crab in a curtain of limbs. Suckers latch onto the crustacean's carapace with delicate precision. The crab thrashes, but the octopus uses its beak, a sharp, parrot-like structure concealed beneath its arms, to deliver the fatal bite. Crabs are evolutionary marvels of defense. Their shells resist crushing, their claws pinch hard, and their joints are reinforced with layers of calcified protein. But octopuses evolved to counter exactly this. Their saliva contains powerful neurotoxins that paralyze prey quickly. Their beaks pierce armor. Their radula, a ribbon of tiny teeth, scrapes through muscle and connective tissue. In some species, specialized glands produce acidic secretions that soften shells or allow them to drill precise holes into snails and clams. The octopus understands its prey intimately, not through thought, but through millions of years of evolutionary refinement. The crab's struggles cease. The octopus retreats into its den, dragging the carcass behind it. Inside, away from the reach of eels or groupers, it consumes the crab piece by piece. The camera cannot follow inside these tight confines, but when the octopus emerges again, its arms glisten with tiny fragments of cartilage and shell. With a sweep of its limbs, it clears its den by pushing the crab's remains outward, adding them to the growing shell heap. This is where the story changes, where the aftermath becomes more ecologically significant than the hunt itself. The shell pile is not inert waste. It becomes a micro-habitat, a structure built entirely from the leftovers of the octopus's meals. At first, it may seem like nothing but a cluster of empty armor. But within hours, bacteria begin colonizing the surfaces, forming slick biofilms that digest the microscopic residues of muscle, hemolymph, and digestive enzymes left on each shell fragment. Underwater, decomposition is swift and efficient. Every scrap of organic tissue becomes food for something. Soon after the bacteria arrive, tiny detritivores follow, small worms, amphipods, and scavenging crustaceans that thrive on decomposing films. They slip into the grooves of broken shells, grazing along edges and interior surfaces. In doing so, they clean the remnants more thoroughly than the octopus ever could, breaking the material down into particles that drift with the current or settle into the sand. These particles nourish filter feeders and contribute to nutrient cycles essential to reef productivity. The camera moves closer, revealing that the shell pile functions like a miniature reef. Gaps between shells create tunnels, ridges, and crevices. Juvenile fish, barely the size of a fingernail, dart between the shards, using the pile as shelter from predators. Small shrimp hide beneath clam halves, navigating the labyrinth for safety. Even larger species, gobies, blennies, and small cardinal fish, begin to treat the shell heap as part of the reef's architecture. What was once a cemetery of armored bodies becomes a thriving community shaped by decay and structure. This transformation highlights a remarkable ecological phenomenon. Octopus feeding behavior inadvertently engineers habitat. Unlike corals that build reefs intentionally, or urchins that clear areas through grazing, the octopus shapes its surroundings unintentionally. It hunts for survival, but in the process it creates habitat for dozens of species. Every meal contributes another piece to the structure. And as the octopus moves dens or grows larger, it leaves behind these enriched zones, which remain productive long after it has departed. The camera pans across the surrounding seafloor to emphasize the contrast. Areas without octopus middens are more uniform. Sand, rock, algae, shells scattered sparsely. Biodiversity there is modest. But around each octopus den, life clusters densely. Predators use the heaps as hunting grounds. Prey species use them as shelter. Algae colonize the uppermost shells, adding color and binding fragments together. Microbial diversity spikes. Even sponges sometimes establish tiny footholds on shell edges if the heap grows large enough. From a distance, each shell mound resembles a pale crater, a subtle indentation in the seafloor marked by shimmering white fragments. But zoomed in, each is a city, multidimensional, constantly changing, and dependent on the octopus's continued maintenance. The film then focuses on the octopus's behavior when selecting shells. Not all shells are discarded, some are repurposed. A broken clam may become a door to the den. A perfectly curved snail shell may be dragged inside for egg protection. An especially large crab carapace may be used as temporary armor during risky forays outside the den. Octopuses are tactile thinkers, evaluating objects with their suckers, tasting, feeling, sizing up potential utility. Each shell has a place, whether as food residue or as a structural element. This dual nature, destruction and construction, is what makes the octopus an essential recycler. It dismantles armored prey that would otherwise remain intact for long periods, turning slow decaying organisms into rapidly available nutrient sources. At the same time, it assembles shell piles that provide long-term benefits to the ecosystem. The narrative widens to consider the ecological ripple effects. By concentrating shells in specific locations, octopuses create hotspots of mineral availability. As shells decompose slowly, calcium leaches into the sediment, influencing local pH levels and enhancing conditions for calcifying organisms. Snail larvae, coral polyps, and tube-building worms benefit from this mineral enrichment. Fish larvae find refuge during their vulnerable early days. Even predators like moray eels or scorpionfish occasionally ambush prey around these heaps, using their structure as cover. The camera dives deeper into one shell heap, showing amphipods scurrying across a clam valve. Bacteria coat the inner surface in subtle, rainbow-sheened layers. A juvenile wrasse pecks at microfauna, hiding between barnacle scars. A small octopus, likely a juvenile from another den, hovers nearby, exploring the edges of the pile. Perhaps one day the youngster will create a midden of its own. The cycle continues. Now the frame draws back to reveal the octopus emerging again. This time it carries a large whelk shell in its arms. The octopus manipulates the shell delicately, rotating it, inspecting its weight and structural integrity. It identifies the weak point, a small notch near the hinge, and slides its beak into position. With a firm bite and twist, it cracks the shell enough to insert its radula. The prey inside is subdued, consumed, and the emptied shell becomes another piece of the growing structure outside. This moment underscores a profound ecological principle. The octopus is both predator and architect. Without intending to, it accelerates decomposition, redistributes nutrients, and constructs microhabitats that increase biodiversity in regions where hard structure is limited. The chapter closes as the camera rises above the boulder field. From the wider perspective, dozens of pale rings mark the seafloor. Each one the footprint of an octopus den, each one a recycling hub, each one a nursery for small species and a feeding ground for larger ones. What began as a field of predators becomes a mosaic of life shaped by predators' leftovers. In the quiet circles of broken armor, life thrives. Rot becomes architecture, Scraps become shelter. And in the shadow of an octopus's den, the armored dead are reborn into an ecosystem built entirely from the remains of yesterday's meals. The shallow seafloor shifts from coral gardens to a broad, quiet plain of sand and rubble. Sunlight flickers across the sediment in wavering patterns, as if the ocean's surface were painting the ground with liquid gold. Broken shells, scattered coral fragments, and smooth pebbles lie partially buried in a delicate layer of silt. It is a peaceful landscape at first glance, a place where the occasional fish glides past and hermit crabs explore abandoned shells. But beneath the stillness, something far more intense is happening. This rubble field is not simply a graveyard of broken things. It is an arena patrolled by one of the ocean's most powerful recyclers. A mantis shrimp rises from its burrow. The creature's eyes are the first part of it to appear. Two periscopes mounted on stalks, rotating with uncanny precision. Each eye is divided into regions that detect light in ways beyond human comprehension. They perceive ultraviolet, polarized light, and subtle patterns invisible to nearly every other animal. These eyes allow the mantis shrimp to read the seafloor as if it were a coded map, identifying prey, predators, and hiding places with hair-trigger accuracy. Its body follows, compact, armored, and brilliantly colored in shades that defy logic. Greens, reds, oranges, and metallic blues that shimmer even in the muted light of the rubble zone. The creature's home is a reinforced burrow, hidden beneath a ridge of broken coral. Its walls are lined with carefully selected shell fragments and stones, arranged in ways that stabilize the tunnel against collapse. The mantis shrimp is not only a hunter, it is an engineer, shaping its environment with obsessive attention to detail. But what it does to the environment while hunting is far more profound. The mantis shrimp emerges fully and begins to patrol. It is a smasher species, equipped with calcified hammers folded beneath its forelimbs. These hammer structures store enormous amounts of elastic energy. When the shrimp strikes, it releases that energy in an explosion of force so intense that the water around the hammer briefly vaporizes into a bubble, a cavitation shockwave that produces temperatures comparable to the surface of the sun for a fraction of a second. The strike does not simply break shells, it obliterates them. The camera follows the predator as it moves across the rubble. Its gait is careful, almost dainty, but each step is calculated. It stops beside a snail creeping slowly across the sand. The snail retreats into its shell, unaware that its defense is meaningless here. The mantis shrimp twists its body, aligns its hammers, and fires. The impact is instantaneous. A deafening crack reverberates through the water. The snail's shell explodes into fragments, dozens, then hundreds. Sharp pieces scatter outward in a conical spray. The shrimp grabs the soft body inside and retreats into its burrow to eat. The broken shell remains on the sand, a constellation of jagged pieces where a single intact snail once stood. This is where the ecological magic begins. Intact shells can persist on the seafloor for years, even decades. They resist erosion, they retain shape, they decay slowly, but fragments weather rapidly. The increased surface area exposes them to currents, grazing detritivores, dissolving acids, and microbial colonization. A shell that might once have persisted for a century can break down in a year once shattered. Mantis shrimp create these fragments at astonishing rates. Shells, crab carapaces, clam valves, coral nodules, all shattered into digestible pieces for the ecosystem. The camera zooms into a freshly shattered shell shard. Bacteria swarm over its edges, forming a thin film of life within minutes of the trauma. Tiny algae spores settle onto it, exploiting the roughened surface. Micrograzers arrive soon after, drawn by chemical cues in the water. They scrape the film, feeding on the newly accessible nutrients. Within days, each fragment becomes a tiny island of productivity on the seafloor. Shattered shells also aerate the sediment. Their irregular shapes trap pockets of water, preventing areas of the seafloor from becoming suffocated by silt. These micro-pockets host worms, copepods, and amphipods, creatures that sift, clean, and oxygenate sediment as they feed. The fragments become scaffolding for small anemones and bryozoans. Some pieces lodge against each other, forming small shelters for juvenile fish and shrimp. In this way, the mantis shrimp, a predator feared for its violent strikes, becomes an unconscious architect of diverse, thriving micro-habitats. The camera shifts to a time-lapse sequence showing how a fragment transforms over weeks. It begins as the sharp remnant of a destroyed shell. Within days, biofilms cover it. Within weeks, algae bind it to other fragments. Within months, the piece becomes unrecognizable, now fused into a patch of sediment enriched with calcium and organic matter. Eventually, the fragment dissolves almost entirely into the seabed, contributing minerals that support the growth of coralline algae and other calcifying organisms. Hard waste that once cluttered the seafloor breaks apart, dissolves, and re-enters the nutrient cycle. But this transformation is not limited to shells. The mantis shrimp also hunts crabs, whose exoskeletons are composed of chitin reinforced with calcium carbonate. These carapaces, once broken, degrade rapidly. They feed detritivores, which in turn feed small fish and filter feeders. On a healthy rubble plain, the remnants of mantis shrimp kills form a mosaic of small habitats, each one an engine of biodiversity. The narrative widens to encompass the larger landscape. The rubble zone extends for hundreds of meters, dotted with burrows, shell piles, and fragment fields. Multiple mantis shrimp occupy territories spaced carefully apart, each individual maintaining a radius around its burrow. The dens themselves, reinforced by accumulated fragments, are structurally stable in ways that pure sand burrows are not. They resist collapse during storms. They protect their occupants and provide shelter to commensal species. The camera reveals one of these dens from within. The mantis shrimp rearranges fragments, hammering some into place, discarding others. It selects pieces of specific shapes to fortify tunnel openings, adding layers of structural complexity. These fragments, once part of intact shells, now form a composite architecture stronger than sand or rubble alone. This engineering enhances habitat integrity, not just for the mantis shrimp, but also for small fish and invertebrates that use the abandoned dens later in life. The ecological impact becomes clear. Mantis shrimp dens are recycling centers. They collect, shatter, process, and redistribute hard waste into forms that the environment can reuse. Without mantis shrimp, many rubble plains would accumulate hard debris that persists for decades, slow to break down and slow to support new microbial or algal growth. But with mantis shrimp, the rubble field becomes dynamic, constantly broken, reformed, and reintegrated into the ecosystem. The camera shifts to another mantis shrimp striking a small clam. The impact sends fragments tumbling through the water column. Some fragments settle immediately. Others drift, rolling across the seafloor until they wedge in cracks or settle into sand valleys. Each piece becomes a seed for new biological interactions. In regions with many mantis shrimp, the density of these fragments increases dramatically, creating patchwork terrains rich in microhabitats. The chapter explores the sensory world of the mantis shrimp next. Its ability to detect prey beneath thin layers of sand means that buried hard waste is constantly unearthed and shattered. It uses vibration sensitivity to locate crabs hiding under rubble. No shell is safe. No fragment remains large for long. The mantis shrimp's relentless need to feed produces constant turnover in the rubble zone, granting the ecosystem a recycling mechanism unmatched in speed or efficiency. As the chapter nears its conclusion, the camera rises above the rubble plain. What first appeared to be a quiet desert reveals itself as a living mosaic, shimmering patches of algae on fragments, tiny fish weaving between shards, shrimp hiding in hollow spaces, worms aerating the sediment around them. These patches stretch outward from mantis shrimp dens, like ripples from a stone dropped into water. The true power of the mantis shrimp lies not only in its strike, but in its unintended legacy, a seafloor where nothing remains solid for long, a world where hard waste becomes the scaffolding of new life, a battlefield where destruction accelerates renewal. What seems like violence is, in ecological terms, acceleration. What seems like waste is raw material. What seems like rubble is the beginning of a micro-reef. The chapter closes as the mantis shrimp disappears back into its burrow, its hammers folding neatly under its body. The rubble plain glows faintly in the filtered sunlight, fragments shimmering, life stirring in every crevice, all made possible by the creature below, the recycler, armed with the fastest strike in the ocean. The chapter opens on a muted, almost colorless stretch of seafloor. Gone are the bright corals, the drifting kelp forests, and the bustling reefs. Instead, a vast plain of fine sediment spreads out in every direction, absorbing sunlight rather than reflecting it. The water carries a faint haze, created by countless silt particles that rise and settle with every passing current. Nothing seems to move at first glance. The mud looks still, layered in silence, holding the imprint of the last creature that passed this way. But this quiet landscape is deceptive. Beneath the top few centimeters lies a world of rot, compression, and chemical tension. Organic debris. Fish remains, decayed algae, shed skin, broken worm tubes accumulate here in slow motion, often trapped in nearly oxygen-poor layers, where decomposition grinds down rather than burns through. The sediment is a memory bank of death, and without constant disturbance, it would become a suffocating sheet of decay. A shadow glides in from above. A stingray approaches the benthos with the calm assurance of a creature perfectly adapted to this muted stage. Its body is a broad, undulating disc, shifting like living silk as it moves. The ray's coloration mirrors the muddy seafloor, a mottled tapestry of browns, greys, and pale sand tones. Its wings ripple in steady motions, giving no hint of the power coiled in its movement. Beneath the disc, hidden from view, is the ray's mouth. A set of grinding plates evolved not for theatrics, but for relentless, methodical feeding. The stingray's eyes, set atop its head like twin sentries, scan the water, while its electroreceptors, clustered in sensory pores called ampullae of Lorenzini, detect faint muscle twitches and heartbeat-level signals buried beneath the mud. The ray circles once, twice, drawing invisible lines in the water, then it stops. Something beneath the surface has called to it, a faint tremor perhaps, or the chemical hint of decay leaking upward. It lowers itself gently, wings easing down until it becomes a seamless part of the seafloor. Silt flows over the edges of its body and rests there, suspended. Then, without warning, the stingray plunges. The entire disc contracts downward as if a force has pressed from above. Mud erupts around the creature in a slow, blooming cloud. A murky explosion of silt, decaying particles, and broken organic scraps. The ray's mouth works frantically beneath the disc, vacuuming sediment, sorting it, rejecting useless grit through spiracles, and swallowing anything edible: worms, buried shrimp, fragments of rotting fish, even tiny scavengers that cluster near decomposing pockets. What looks like a simple feeding technique is actually a powerful ecological service. By disturbing the sediment, stingrays release pockets of trapped nutrients that have settled invisibly into the lower layers. They expose buried carcass fragments that might otherwise remain sealed in anaerobic mud, where decomposition is slow and toxic gases accumulate. Each plunge oxygenates the upper sediment layer and interrupts the formation of long-term anoxic zones, pockets where rot becomes locked away, producing hydrogen sulfide and methane that poison the benthos if allowed to expand unchecked. The ray rises from its crater in a smooth upward lift. The mud plume continues to expand, drifting outward like a nebula. Small fish appear almost instantly. Gobies dart through the cloud, snatching newly exposed worms. Juvenile jacks flash in and out, picking off tiny amphipods tumbling in the water. Even hermit crabs sense the disturbance and scuttle toward the plume, guided by the chemical signature of decaying proteins suddenly freed from burial. Instead of a monotone desert, the mud plain becomes a momentary feeding frenzy, a pulse of life triggered by the stingray's simple act of digging. The camera sweeps across the seafloor to reveal dozens of such craters at varying levels of freshness. Some are crisp-edged, newly formed, others are soft, partially filled, weathered by currents. These pits form a mosaic of oxygenated zones, each one preventing the sediment from becoming a sealed, stagnant layer. Without stingrays, this plain would grow stratified in chemical gradients. Oxygen-rich at the top, lethal below. But the rays constantly plow it up, tossing rot back into circulation, making death edible again. As the ray glides to its next target, the narrative shifts to examine its feeding mechanics more closely. The ray pulls its wings upward slightly, creating a pressure differential. Water rushes beneath, carrying sediment into its mouth. The grinding plates sort by density. Heavier fragments such as small shells and bones are pushed aside. Softer materials, buried mussels, bits of dead fish, decomposing plant fibers are consumed. Tiny organisms living within the rot become unintended prey. This process is more than foraging. It is sediment turnover at a scale few predators can match. Where crustaceans pry and pick at exposed carcasses, and worms slowly feed on buried detritus, stingrays operate in a different dimension. They do not wait for rot to reach the surface, they unearth it. The camera transitions to a close-up of the sediment layers. The upper centimeter is light and oxygen-rich. Below that, a darker layer begins. Compressed, low-oxygen mud where anaerobic bacteria thrive, releasing gases as they slowly break down buried organic matter. Deeper still, shells and bones sit half-dissolved or nearly intact, depending on their age. When stingrays dig, they churn these layers, exposing trapped carbon, nitrogen, and phosphorus back into the active biological loop. A second ray joins the first. Their territories overlap loosely, not in strict boundaries. In areas where stingrays congregate, the sediment looks almost cultivated, soft, aerated, stirred frequently. The landscape resembles a farm field turned over by invisible plows, each crater representing a disturbance that resets the decomposition process. This plowed terrain supports far more surface-dwelling microbes than unturned mud does. Microbial films flourish on newly oxygenated substrates. Filter feeders in the water above respond quickly to the increased production of organic particles. Bird's-eye footage reveals an entire section of seafloor shimmering in various shades of brown and grey. The marks of countless feeding events over weeks and months. A field of craters covers the expanse, proof of sustained disturbance. In every depression, small scavengers cluster, cleaning up what the ray missed. A buried fish corpse offers a vivid example. The camera isolates a partially decayed mullet trapped a few centimeters beneath the silt. Bacteria coat its skin in a slimy film. Gas pockets bubble softly around it, trapped by the weight of the overlying mud. Left alone, the fish would decompose slowly, releasing toxins that spread horizontally through the anaerobic layer. But the stingray senses the faint chemical ghost above it and dives. Its body hits the ground, wings folding down like a shroud. Silt explodes. The corpse emerges in pieces, and a swarm of small predators moves in to help dismantle the remains before they can form a dangerous, expanding zone of rot. In a matter of minutes, a hidden reservoir of decay becomes a feast for dozens of species. Once again, a predator has changed the chemical trajectory of the seafloor simply by feeding. Stingrays also assist sediment health through repeated visitation. A single area may be dug out several times in a week, preventing reburial and stagnation. Their foraging pits increase surface roughness, a key factor influencing microbial turnover, water mixing, and the settlement of larvae. The pits create gradients of depth and shadow where some organisms hide and others forage. They transform dull uniformity into patchwork heterogeneity, which increases biodiversity. The cinematic narrative zooms outward to describe nighttime behavior. When darkness falls, certain stingray species become even more active. Their electroreception guides them with uncanny precision, allowing them to sense the faint discharge of hidden fish beneath thin sediment layers. They plunge repeatedly, digging trenches rather than isolated craters. At night, the plain becomes alive with suspended particles, drifting across the landscape like underwater dust storms. These disturbances do more than expose prey. They prevent the seafloor from becoming a static sink where dead matter accumulates faster than it can be processed. In essence, stingrays act as rot regulators. They convert compressed, hidden decay into accessible food for smaller scavengers and into oxygenated sediment suitable for worms and microbes involved in deeper, healthier nutrient cycling. As the narrative approaches its final sequence, the camera reveals the broader ecological consequences. Areas frequently plowed by stingrays host healthier communities of benthic organisms. There are more filter feeders because more particles stay suspended. There are more juvenile fish because the craters offer shelter and feeding grounds. There are more worms and small crustaceans because oxygen penetrates deeper into the mud. Even the chemical composition of the water above these plains changes subtly. More dissolved oxygen, fewer sulfide-rich plumes, more suspended organic particles available for plankton. These minuscule shifts scale up dramatically across coastal regions where stingrays are common, altering local productivity in ways scientists are only now quantifying. The chapter closes with a final sweeping shot, a stingray gliding effortlessly over a field of its own making. It dips once more, stirring a billowing cloud of sediment that twists upward before dissolving into the surrounding water. Small fish rush in behind it. The crater forms like a fresh scar on the seafloor, but one already teeming with opportunity. This is not a graveyard, not a stagnant plain, not a dead zone waiting to spread. Thanks to stingrays, the mud is alive, constantly plowed, constantly oxygenated, constantly fed into the open circuit of life, rather than locked in the slow suffocation of buried decay. They are gardeners of rot, turning death back into motion, back into energy, back into the chain of predators that will rise and fall above them. The seafloor ends the documentary not as a silent tomb, but as a field shaped by sweeping wings. A living landscape maintained by a predator who feeds by plowing, restores by disturbing, and keeps rot from sleeping too long below the world's quietest plains.